that we will take to her later to indicate what we did during this symposium. So this symposium is to celebrate Mary Lou's 100th birthday. And it's 100 years of her living in Nebraska and her work at the university and the University of Nebraska State Museum. So welcome to the Centennial Symposium honoring Mary Lou Hanson Pritchard, who turns 100 years old today. So Dan and I planned this symposium to honor not only the work that Mary Lou has done in the 1900s and 2000s to bring the science of parasitology to the Great Plains of Central North America, but also to honor her dedication to training women in science. Thus, we are honored to have young women speak today and tomorrow in this symposium. Some of my students and some other students will be here speaking of their current research based on collections of specimens in the field and using collections material that is in museums. Mary Lou was able to break many of the so-called glass ceilings that non-enlightened scientists had established in science with her firm guidance and direction. Both Dan and I have been able to continue her work. Although Dan's retired, I'm still forging ahead. We'll see if, and Dan's not really retired, he just acts like it sometimes. So Mary Lou brought the ideas of parasitology along in an unbroken lineage, first starting with Leuchart in Germany, where Henry Baldwin Ward trained, and then Franklin Barker. Well, Henry Baldwin Ward trained, then came here, then he trained Franklin Barker, who was here for several years, and then Harold W. Manter took over at that point after Barker retired, and Manter was here from 1925 till he died in 1971. And then Mary Lou Pritchard. Mary Lou Pritchard, well, I was lucky to have arrived in Nebraska just as the curator, Mary Lou Pritchard, had retired. And she was a curator in the University of Nebraska State Museum Division of Parasitology. She started the division in 1971 based on Dr. Manter's nidus, based on his main collection of parasites, mostly from marine fishes. But he did a lot of other things besides marine fishes. Mary Lou retired from her duties as curator of parasites at the University of Nebraska State Museum in 1993. I started in 1994. She then became curator emerita and the Manter Laboratory of Parasitology librarian. And we really needed a librarian because she accumulated so much material from all over the world that it's important for us to keep that library going. It's really the biggest parasitology systematics resource library in existence. Soon after her retirement, Mary Lou was given an honorary doctorate of science at the University of Nebraska, Lincoln, and she continued her duties as librarian and emerita curator. Mary Lou has shown unflagging support for her beloved Lincoln and Nebraska, and she single-handedly brought the collection of parasites that Dr. Manter had gifted to the State Museum, to the Division of Parasitology here at the university, up to the second largest collection of parasites in the Western Hemisphere. With the support of the global community of parasitologists, we have recently edged out the former U.S. National Parasitology Collection. This collection is now distributed not as one unit any longer throughout the Smithsonian. It's no longer a powerhouse national collection that it used to be. So we've now surpassed the size of the former U.S. National Parasite Collection with the addition of several 100,000 specimens of fleas and other ectoparasites from people all over North America. We are now fourth or fifth largest parasite collection in the world that we house over in Nebraska Hall. Due to the initial efforts of Mary Lou, the HWML parasite collection is now fully digitized and geo-referenced. With all parasite data available via the Global Biodiversity Information Facility, GBIF. It's not well known that Mary Lou enabled digitization of biological collections in North America, bringing collections in museums into the computer age, first by putting the parasite collection of the Mantra Lab into the data analysis program SAS. And that was done before there were microcomputers or mini computers. It was stored on a mainframe. And then after mini computers and microcomputers came out, she bought a 286 with 100 megabytes of space and transferred all of the data from the SAS program onto the RBase 2000 program. The RBase 2000 program that she used was distributed all over the United States, which basically started the revolution of microcomputers entering museums' collections. And so what she did was she stripped off the data from our database, but she shared the template of the RBase 2000 all over the United States during that time. So UC Davis had one. It was done at University of Georgia. Everybody used that same system. But then, of course, it then was modified into DBase 2, then DBase 3. And then now we're moving forward into other more difficult to understand databases. Her teaching and guidance of master's and doctoral students established in the establishment of the Mantra Laboratory of Parasitology after Mantra's death in 1971, and her unwavering dedication to the University of Nebraska and to the University of Nebraska State Museum are unparalleled. Nobody else has put that much effort into this system in Nebraska at the University of Nebraska. So this symposium that Dan actually came up with, and I agreed with wholeheartedly until he almost died. And then I said, I don't know what we're going to do, but then he came back. So here we are. The symposium is dedicated to Mary Lou Pritchard to honor her and to celebrate her 100th birthday. If anybody would like to submit a letter of greeting, letter of compliments, an interesting story, or anything like that, please send me an email with that information in a Word file, and we will compile it in really large type, and we will then send it to Mary Lou. I will take it to her house, and we will then present it to her so she can actually have someone read it to her, or she can actually try to read it herself. But she can just not see well enough or hear well enough to do it on her own. We will format and compile the letters and print them for her, and I can make them available on our web server also. So if anybody has anything like that, please accumulate it, put it together, get it to me. So I'm going to basically, we have 15 more minutes of introductory statements, and I think we can just start now if Dan wants to begin. If anybody has any comments at this point or any discussion about how databases first started in the US, the Mantra Lab database is now running in one main instance, which is called Arctos. And then we have a sub instance called running on Microsoft Access, which we have as a backup in case the Arctos system goes down, which it does all the time. And so we have our system based on Arctos, which is a distributed database system with lots of different collections in it. And also we have a Microsoft Access database, so we can more easily run the material and use the material in our collection daily. Our collection is now over, what's the size now? 285,000 catalog records? Yeah, about 200,000 actually catalog records. And that means one catalog record could actually have 500 individuals in that catalog record. So we really have several million individuals in our collection. So, all right, I'm going to turn it over now to Dan. Dan can start. I think it's okay to go a little early. We have lots of people from the internet here. Walter from Brazil, Tom Levette's here in Lincoln, Tim Yoshino, Tom Platt, John Shea. I don't know if everybody can see each other. Susanna von Essen. And so we have a lot of distinguished guests with us here also. I think, do we have a video of the front here too, or is, all right, okay, good. All right, so let's let Dan begin to speak, let me put up your PowerPoint. It should be working here. This one?
No, it's the intro.
This
Mary
one?
Lou intro.
The other left. Look okay, Gabor? No, but I will. This should be it. And it looks okay. There, doesn't look the same here, but it should be okay. Let me move these out of the way.
Presented.
Got it? So Dan Brooks, one of Mary Lou's students. Dan was an undergraduate here. You're probably gonna say all that?
No.
Dan was an undergraduate here at the University of Nebraska, Lincoln. He was recruited from high school to be a high jumper here at the university on the track team. And so he was a star track a high jumper, and he actually was, it was the last year when he finished up when the Fosbury flop came in. And so he was
Put me out of business.
he was a classical jumper over the front, and Fosbury went backwards and it put him out of business because he didn't do the flop. But then Dan went on, finished his PhD with Robin Overstreet, who just expired this year. Robin Overstreet was down at Mississippi, and Dan worked at Gulf Coast Research Lab. So I'll let you go.
Super. Thank you very much. Thank you, Scott. Thank you, everybody online and here for coming to celebrate Mary Lou Pritchard's 100th birthday. Now, I don't promise not to get a little bit emotional at times through this, but I'll try to get through it anyway. Oops, that's not. What do I do to advance this?
Well, maybe use this page up.
There we go.
Maybe page down.
No.
Okay, we can go like this. Maybe use this thing, or maybe arrow key will work. Yes,
Okay,
arrows.
all right,
Use
all right.
those arrows.
Okay. So I'm going to talk just very, very quickly. A lot of the information I'm going to go through quickly now is also online at the website. But I want to emphasize a bit about just how accomplished Mary Lou was as a student and as a scholar. And to put that in the context of what she had to put up with being a pioneering woman in science at the time. Now let's see if I can, okay. So in 1942, Mary Lou entered the University of Nebraska as a freshman. And in 1946, at the spring convocation of the University of Nebraska, she received a Bachelor of Science degree with distinction in business administration. That's what the record would say, but that's covers up an enormous amount of what she actually did. So for example, she majored in business administration, accounting, French, and zoology at the same time. She had academic scholarships every year, including one of the prestigious Regents of the University of Nebraska scholarships. And even that doesn't tell the story, because during that time, she was elected to the National Romance Languages Honorary Society, the National Business Administration Honorary Society, the National Business Administration Women's Society, for which she won the 1946 Key as the Outstanding Woman Graduate in Business Administration at Nebraska. And of course, she was a Phi Beta Kappa. Now this is, to use a sports analogy, this would be the same as somebody being an All-American in football, basketball, and baseball in the same year, and then going to the Olympics and winning the modern heptathlon, all in 1 year, or in a 4-year period. And I would be willing to bet that she is the most decorated, academically decorated undergraduate in the history of this university. If she isn't, I would really like to know who did more than this. In 1949, she graduated with a Master of Science degree in zoology. And once again, that doesn't really tell the story, because she received, during that time, she received every year graduate fellowships in both biology and business administration. At the end of her undergraduate year, she won a prize, and she used that money to do the field work for her master's degree, which was parasites of fishes of Hawaii. She would, at that time, so this is the late 1940s, she lived in a little pension at one end of Waikiki Beach. She would get up every morning and she would walk the length of Waikiki Beach to a little coffee shop where she'd have coffee and a pastry. She would go to the fish market and get her specimens. She would walk back to the lab and she would work all day long. And she said she was the only person on Waikiki Beach at that time. So, I mean, this will never happen again, right? So the interesting thing, oh, I'm sorry. Oops. Where are we back? Okay. Yeah. She also won the Wolcott Prize as the Outstanding Zoology Graduate Student. She became a charter member of what was then called the Society for Systematic Zoology. It's now the Society of Systematic Biology. She may be, I'm not certain, but it wouldn't surprise me, she may be the last surviving charter member of that society. And she was elected to Sigma Xi, National Science Honorary Society, during that time. But then she hit a roadblock. So after all of that undergraduate and graduate accomplishment, the chairman of the Department of Zoology at the University of Nebraska told her, it is our policy not to recommend women for PhD programs. And that was effectively it, except they were dealing with Mary Lou Pritchard. And she just simply said, okay, that's a roadblock, I'm going to go around it. So in 1951, she began working as a research associate with Harold Manter. In 1959, she was elected to Sigma Delta Epsilon, the Graduate Women in Science Honorary Society. In 1971, as Scott said, she became the founding curator of the Manter Lab. And one of the things that a lot of people don't know is that between 1976 and 1980, she served as a consultant for what was then a fairly new organization called the Environmental Protection Agency. And this reflected her interest in the natural history and biodiversity of the state of Nebraska, which is one of the themes that we're going to explore this next 3 days. Now, once she became the curator, and many of the things that Scott talked about, again, there's a parallel between some personal recognition and recognition for the lab. This is always, none of this was ever done in a very self-serving way. So in 1980, she was granted tenure and promoted to full professor at the University of Nebraska, even though she'd not been granted a doctoral degree. And in 1994, she was awarded the President's Special Award for Distinguished Service by the American Society of Parasitologists. And at the same time, in 1981, for example, the lab was designated one of 4 national resource centers for parasitology by the American Society of Parasitologists. And by 1994, it had already become the 6th largest parasitology collection in the world. And Scott has continued that tradition and exceeded that. So now we have, and yet she persisted 2.0. She finally got a doctoral degree in 2003 when the University of Nebraska granted her a Doctor of Science degree. And I'm now going to do This is the only time we're going to do any sort of personal reminiscences and things. But I'm going to take speaker's privilege and I'm going to just quickly talk about 4 things that have happened to me in my career after I left Nebraska, after I started my own academic career, that give you a flavor of how quietly and pervasively Mary Lou's influence shows up. So in 1986, I was invited to speak in a symposium at the International Congress of Parasitology in Brisbane, Australia. Now, at that time, I was doing something very, very risky. I was advocating that parasitologists do something called phylogenetic analysis. And at that time, anybody who was doing that was putting his own career in jeopardy. That people you have no idea how controversial this was at the time. And so I gave my talk, and a very prominent parasitologist from Great Britain, H.H. Williams, stood up at the end of my talk, turned his back to me, and then lectured the audience about the evils of phylogenetic analysis. And he ended up with a very amusing anecdote comparing my publications with used toilet paper. And then we broke for coffee. And we just, I just walked off the stage, and a number of my colleagues came up and said, "Why didn't you go after him? He was really horrible to you. It was very unprofessional. You should get after him," and all that. And I just went like this. Mary Lou came up to me, patted me on the arm, and said, "That was exactly the right thing to do." In 1993, Eric Hoberg and I were invited to the Natural History Museum in Geneva, Switzerland, to participate in what became this group of people working on tapeworm systematics, really what's become a big collaborative group that's done incredible things. And at that time, so this is 7 years later, so parasitology was starting to say, well, maybe there's something in this phylogenetic analysis stuff. So they asked me to give a talk about principles of phylogenetic systematics in the museum to these parasitologists. And the day it was happening, one of the curators came up and said, "There are all these people, these other curators and other disciplines in the museum that want to come and listen to your talk, and we don't know why they're doing this." And Eric and I just laughed, because in fact, other disciplines had sort of gotten the word about phylogenetics earlier than parasitology. So I gave my talk, blah, 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 blah. And afterwards, a woman who was Chilean by birth, working at the Natural History Museum in Geneva, came up to me and she said, "I was on a National Geographic research vessel off the coast of Chile, and there was a woman parasitologist on that boat." He said, "She was so nice, and she just worked all the time. And she still sends me Christmas cards." And I said, "Mary Lou Pritchard?" She said, "Yes." And I said, "I did my master's degree there." And she went, "Oh, wow. Okay, this is very good." And then 3 years later, I was doing some teaching and some speaking in South Africa. This was the year that apartheid ended and Nelson Mandela became president. And I gave a talk to the Royal Society chapter in Cape Town. At the end of my talk, this elderly couple came up to me and said, "Well, I noticed in the introduction that they said something about the University of Nebraska. There was a woman from the University of Nebraska who was here many years ago. She was so nice, and she just worked so hard. She worked all the time, but she was really nice, and she still sends us Christmas cards, and she sends the gentleman, the husband, was a stamp collector. She sends new stamps from the United States to me all the time." I said, "Oh, Mary Lou Pritchard." She was Mary Lou Hanson at the time. And they said, "Oh, yes, I did my master's degree there." "Oh, well, that explains a lot." And then finally, just before in 2019, I was invited back to University of Nebraska to speak in an event honoring the 50th anniversary of something called the Centennial Education Program, or Centennial College, which was a residential college experimental sort of learning thing. And I was in that program, and in the course of the events, I met a retired senior administrator from the University of Nebraska. And I said, why why was Centennial College canceled? And he said, well, we wanted to expand the engineering program at the university, and we didn't, and we had to find the money for it. So the state legislature wasn't, they mandated that we expand engineering, but they didn't give us more money to do that. So we had to find it. So we started going around, taking money from as many programs as we could. He said, and the only place, and you could tell, I mean, his whole body language changed. This was many, many years later, he's retired. His whole body language changed. He said, there was this one woman in the museum, and she was just completely persistent and implacable. Said, and we knew that if we cut anything from the museum and we didn't give her at least some of what she wanted, she was just going to keep coming at us and at us and at us. And I said, Mary Lou Pritchard. And he said, yes. And I said, yeah, I understand. I understand. And so happy birthday, Mary Lou. We love you. We have great affection for you. We respect you immensely, and we have great regard for you. And now what we're going to have for the rest of the 3 days is a series of lectures that focus on 3 areas that Mary Lou was most interested in. One is the importance of museum-based science. One is opportunities for women, young women in science. And one is the future of the state of Nebraska, which she loves and which she has been tremendously loyal for her entire life. And I look forward to hearing these presentations by these amazing people who've agreed to come and talk. Thank you.
Okay, so it's 9:30. I think we have right on time. We can get moving with the first talk. Let's see here where we are with this one. Should work. Let's see what we have for my ability to get into this.
You have to put another thumb drive.
I think I put it on the desktop. It should work. Let's see. I will close yours. This one?
No. Results.
Where are we?
GPS.
Yes. Got it. Open. Share. And we should have it working. Should be ready to go. So Griselda Pulido Flores is going to speak next. She's a professor at University in Hidalgo, at UAEH, and she has been working on parasitology for the last 30 years. And she's one of the senior research fellows, research associates, or research fellows in the University of Nebraska State Museum Division of Parasitology. And she works on monitoring parasites and other kinds of parasites too. And so we've known each other for, geez, since we were graduate students, I think. So a very long time. And so we're really honored to have Griselda here from Mexico. They came all the way up, Scott Monks and Griselda came up, and they're here doing some work in the museum also. That yesterday they were in the museum all day looking at specimens. And so Griselda will speak Women in Science and Research at Natural Science Collections. Thanks for being here.
Take your phone, I can.
Yes. Oh, share? How do I do that, Gabor? Yeah, I'm trying to find it. Here, I'll go like this. Go back to Zoom. Go to, don't see my share button. See it? should be on here. Got it. That one. Okay. I don't know why it stopped. Excellent, thank you. Yep.
Hello, everybody. For me, it's I'm glad to be here, and it's a special time to celebrate Marilu. When Scott sent me a message if I can come and give a talk about the museums and the collection, of course I say yes, I will be very glad to be. So thank you for inviting me, and I'm so glad to be part of the celebration and give an honor to Marilu Pritchard. So today I'm going to talk about women in science and in research at the Natural Science Collections.
Arrow key should work.
This one?
Yeah. Not yet. Let's see. Let's move this down here. There you go.
I can move by that.
Yeah, try that.
Okay.
Then this should work.
Yeah. Okay. Perfect. Thank you. Well, everybody knows that there are many museums around the world, and all of them are unique, and there are, of course, a lot of collections. And here are some of the most important museum collection, at least in zoology and parasite animals. In America or in United States, we have the Smithsonian, the American Museum of Natural History, of course, the Mantel Lab, that is, we were listening something that there is a collection that some of the most important about parasites. And in Mexico, we have the collections of the UNAM. UNAM is a part of the research center, part of the UNAM, the most bigger university. And of course, we have also some collections and museum in Pio Cruz, that is the Oswaldo Cruz in Brazil. This is in America. But of course, in Europe, we have more collections and museums that they are, of course, more older than in America. And the most important, at least for parasites, are Natural History Museum from London. Of course, there is the Museum of Berlin, and France is very important too, because we have the Museum of National History. And in Spain, we have a Museo Nacional de Ciencias Naturales. And all of these museums have a lot of specimens and a lot of information about the biodiversity of the world. And in Africa, of course, there's a lot. Just I have one here at the moment. There's the Natural History Museum of Zimbabwe. And for the parasitology persons, one of the international museum that is very important for us, and maybe everybody knows, is about the Meguro Parasitology Museum. This museum is very important because have a lot of samples from different kind of parasites from around the world. So as I said, all of the collection and museum are unique, and they have the unique information that they are very important. However, all of them share similarities. The principal goal of the museums or the collections are conservation of samples. Also, the museums are collections. Another goal is cataloging samples data, handling and transportation, different samples, and of course, digitizing and documenting all samples and information that they have or they have. For example, we have here in these pictures in Europe, in Spain, in el Museo Nacional de Ciencias Naturales in Spain, Some women working in this kind of the activity. In all of these activities, the women are working around of the world. In Spain, one are cataloging and another woman is working with some records about some data from the museum. Also, in the National History Museum in London, some women are digitizing and documenting some samples and informations. But also women work and conducted some another studies on the field. For example, here I show you some of my students that they were collecting some rodents in the middle area of Mexico, in Hidalgo state. And of course, we collecting tape, I forget the word in English, renacuajos, tape poles that, because we were looking for some digenics, some larvae of digenics that they are parasite for some frogs. And of course, this is very important for documenting the biodiversity of any place around the world. This is a kind of the sample. Also, women conducting different kind of the projects about biodiversity inventories. Here, I am showing you two species from monogenes that they are parasite from the stingrays. The first one is Dendromonocotyle octodiscus, is a parasite from skin. I collected them in Caribbean, in Atlantic, in Mexico. And the sister group of this species is Dendromonocotyle cortesi, and it's a parasite that was collected by another woman, my teacher, La Maestra Bravo Hollis. She collected this from a stingray in El Mar de Cortés in the Ocean Pacific. And of course, I am collecting some samples from stingrays. I am very interested about biodiversity inventories. But because there are another kind of studies, some women conducting genetic studies of parasites and hosts. In this case, Doctora Christian Bautista, she was collecting the genetic parasites from freshwater fishes in some rivers from Mexico and tried to find how was the genetic and the correlationship, all of this information. Also, at the museums or the labs, women do dissections, processing samples and specimens, and also we collected data at the museums or on the field. Doesn't matter what kind of the place we are going, we can collect every kind of data, and they are very important for the research. Of course, we do some drawings, we recording, as I said, and analyze some data. Also, we do some illustrations, scientific illustrations, and of course, we publish some research, in my case, in taxonomy, systematic ecology, and biography from parasites from vertebrates. But the most important for me are marine fishes, parasites from marine fishes. I prefer them, I love them. So, but all of these activities we do as a woman equals as a man in the world, around the world. And we participate in all of the collection, in all of the museums. Also women conducting different kind of the meeting as a workshop, as a symposium, and also we are teaching and training different students at the university and the research centers around the world. So as I said, women can do anything equals as man, in any collection of any museums around the world. Now, today, we have manager collection, women manager collection. We have here Anna Phillips. She is the curator of the National Museum, the Natural History from Parasites. And also, we have Helen Hardy. She is a digital program manager in the National History Museum in London. And in general, all women believe that if we share the data from the collections, we can transform the scientific research and help find solutions for nature and from nature. Also, we believe that the research comes by sharing our specimen is an immense, has an immense potential to transform our world and help both people and the planet. And this is very important. For example, Scott was talking that yesterday we were working at the museum at the lab with some samples. And of course, it was possible because Smithsonian sent one lot to the Banter lab, and it's the way that I can access to these worms and work. We do some research from parasites in America, some parasites from frogs. So this is very important, and if the collection can share the information of the data and the sample, we can do too much. But all of these activities, as I said, women are working around the world, equals as a man. But in the past, the recognize for the women didn't was enough, or sometimes they didn't get recognized. The first, I'm going to talk about a brief history about the women in science, and that I believe that is important to know. Elena Lucrezia Cornado was an Italian philosopher, and she was the first woman to get the doctor of philosophy, and she worked in mathematics at the University of Padua, or teach at the University of Padua. The second woman to get the doctoral degree, but the first doctor in science, was Laura Maria Catarini Bassi. She works in physics, mathematics, and biology in Bologna University in Italy. We have here another naturalist woman that worked with biology. The principal area was botany and entomology. She was Ana Maria Sibila Merian. She crossed the Atlantic from Amsterdam to Suriname, and she wrote a series of book series that was published in 1060-75. In general, the person now actually, or today, recognize that she was an inspiration to Carl Linneo, that we know he was the father of the taxonomy. She also do a lot of illustration from, there are here some kind of illustration that she did in her time. We have here Dorothea Milova Alice. She was the first woman scientific employed by the Natural History Museum in London. She worked with archaeology, and she was interested in historical ecology. And although she was the first woman with employed by the museum, the employee was unofficial. She was only paid for piece of work. That means that she get only money for one piece of at the time. So this is terrible. In general, as I said in the past, women didn't get enough recognized about the job. We have here another woman that worked in illustration. She was Elizabeth Tiny, and she is the famous Dynasty family, and she do a lot of illustration, principally from flowers and fruit, and works, of course, for the Natural History Museum. Here we have Lucy Evelyn Chessman. She was an English entomologist, and she did an expedition and crossed the South Pacific Island, but she was rejected by veterinary school just because she was a woman. That was terrible. But she, as Marilu, she pushed and she continued working, and after time, she was eventually assistant curator of the insect in the London Zoo. Also, she published more or less 16 books about the insects. And we have here about the museums at the collection, Grace Edward. She was a skilled scientific artist and models maker at the Natural History Museum. And we are at the museum right now, and you can see different kinds of the models. And we can see that they are very, very important to teach the students or to teach the people in general how they are different kinds of the samples. So she was working with this kind of the work at the museums. She prepared illustration and models, but also she painted some buckskin flies by watercolors. And this is in the Natural History Museum in London. Well, in all of these activities, as you can see, women were working at the museums, at the collection, but as I said, in general, they didn't get enough recognized for the work that they did. Also, as a scientific, in general, in the past, women have often been overlooked. That means that, for example, well, I'm going to show or talk about Jane Golden. She was, although she didn't work at museum, she was the first woman in the United States, was distinguished as the botanist. However, she never got recognized for partners in botany at the time. Although she was working and applying the linear system of the classification. The principal area that she was working in America was in New York, and she wrote papers and describe and illustrate about more than 300 species of plants. Unfortunately, this work never was published. So this is so sad. As a parasitology, and all of you that if you are a parasitology, you know very well who is this person. This person was Saiju Yamaguchi. He was a Japanese. He was a very productive person and worked with different kinds of parasites from vertebrates. And he wrote many papers, and everybody knows that, maybe knows him by the system Helmi, that is a kind of series of books about the taxonomic key and systematic ideas or relationship ideas that he had about different kinds of parasites from vertebrates. In this case, he wrote about trematodes with the genetics, monogenes, cestoda, nematoda, and acanthocephala book. and all of them are any kind of persons that they are parasitology know that I am talking right now. And we can see, I don't know if that is enough, but although she didn't get recognized, all of this system helping, he got the assistance from Ikuko Yamaguchi. She was, she typed the information. Also, she made the drawings of in general, all the publications from Yamaguchi. And also, she helped a lot, was an assistant for him for all of the papers. I tried to put in yellow that Yamaguchi recognized, Saito Yamaguchi recognized the work from Ikuko Yamaguchi. Here are one sample, the kind of the drawings that Ikuko Yamaguchi made for the papers from Saito Yamaguchi, or for the Sistema Elmington that I was talking. If you can see, the details are wonderful, and as a taxonomy, they are very, very important to know and can find difference between one species and another species around the world. And also in yellow, I mark that Yamaguchi recognized her work. Here, the first time to the talk, I say something, I talk about the Meguro Parasitology Museum, that I say that this is a very important museum for all parasitology persons. Well, in this museum, I know that there is a place, or they have a stand with some work of Yamaguchi, Saito Yamaguchi. The museum recognized his job. Unfortunately, they don't recognize, or they don't mention any about Ikuko Yamaguchi, and she made all of this, in general, all of this drawing. So this is so sad. It is good right now, or today, that we can, as a woman, we can get recognized from our work in collections and in museums. And now, of course, I'm going to talk about Marilu Hanson Pritchard. As Dan and Scott told us, she established the Harold W. Manter Laboratory of Parasitology, and we know now that there is one of the most important collections of parasites in America. She did that in honor to Harold Winfrey Manter. He was a colleague from her. She worked with him with some parasite from marine fishes. And also she was member of the ESP and she was reviewer, consultant, and assistant editor of the Journal of Parasitology, and many other things that I don't need to pronounce or say, because of course Dr. Dan was talking. But something that she did, and that is very important from the collection, that is that she was thinking as a scientific at the time. She coordinated and growing the parasite collection, the Manter Lab, also developed the access record systems, and she made a lot of notebooks with information of different kinds of parasites. Scott was talking about all of the program and all of the catalog number that she started, and now he's continuing with this information about that museum. And here in the pictures, we have, I asked Scott, help me with this, but I want to show you all of the notebooks that Marilu did with the idea from try to classify the parasites, but also to help students and other persons that they were doing research at the time. Here is an example, a kind of the notebook that some degenics that Marilu did. And if you see, they were carpets or notebooks with different information about the different papers, and they put together as a family. And this was very easily, if we want to consult one information about the some family from parasites, it's very easily try to follow the information with this kind of the notebooks. Also, Marilu started the library records by the authors. And for me, I want to share you that I met her about 25, 26 years ago, the first time when I came Lincoln and visit the university. And she received me very friendly, and she told me anything that you need, you can look for any information that you need, and you are looking, maybe you can find here at the library. And I need to say that without this information, without all of the job that Marilu did before, I couldn't write my PhD thesis without all of this information, because if not, it was so difficult try to look for by the papers or books or illustration from different kind of the parasite that I was working around the world. So it is so difficult. And thanks to Marilu, Pritchard was very easy to me, get information, and I can do very well, very easily, more or less, write my thesis of PhD. So thank you, Marilu. And of course, Marilu was training, she was teaching. I don't know exactly how many students she got and she teach, but I know at least that two of the most distinguished students were or are Dr. Brooks and Dr. Janine Kaira. They were students from Marilu. So they have the ideas and the influence from Marilu, and I believe that that is a very good work from Marilu. Okay, here is Master Margarita Bravo Hollis. She was a colleague from Marilu, Dr. Marilu, and Maestra Bravo is, as we call in Mexico, Maestra means teacher. She was working at the Colección Nacional del Mitos, that is the most important collection of parasites, at least of the helminths in Mexico. And she with Dr. Caballero worked at the museum and worked at the collection, cataloging different kinds of the samples in their times. Here are some the new buildings of the museum, but the first time was in Chapultepec. I don't know if that some of you know Mexico City, but Mexico City, there is a very big area in Chapultepec. It's a very traditional area, and it was the first place by the museum in La Casa del Lago. It's a big lake, and that was the first museum, but now it's at the south of the Mexico City in UNAM. Okay, the Maestra Margarita Bravo, she was a curator of the mythological collection at the UNAM between 1960 and 1980. She published the first catalog of the collection, and she was a parasitology person, but the principal group that she was working were monogenes, principal marine fishes, and she described more or less more than 100 species, and also she wrote more than 19 papers. She was, as I said, she was a colleague from Marilu, and I know that she came to Manter lab many years ago. I don't have the date, but I know that they were working together and teach some students. Well, now in Mexico, in general, and in helminthology, the person recognized 5 persons that we are the top helminthology doing research. The first one, of course, is Maestra Margarita Bravo Follis. She was a mentor from us. In this case, we have here Doctora Virginia León Regañón. I know that some of you know her very well. She worked at Ibunam. Also, we have Rosario Mata Lopez and master degree Berenice Mendoza Garfias. All of them are working at the UNAM in a different areas at the UNAM. One of them in el Instituto, and the other is working in Facultad de Ciencias. Well, I am the number 6, more younger, of course. So, and now although I studied my master and my bachelor at UNAM, now I work in a different university. Now I work Universidad Autónoma del Estado de Hidalgo. So, and now I would like to show you some of my students with some colleagues. Of course, he is Scott Monks and Dr. Jorge Falcón. Well, something that I need to say and try to be remarkable is that actually, or today, women can do anything equals as a man in collection and museum. Unfortunately, in the past, all of this work in general, the women didn't get recognized. We know the history about Marilu, and sometimes the persons don't recognize or didn't recognize his job, her job. Thank you.
Any questions or comments? Anything from the internet? Do you see got more? Okay.
What town in Mexico? What city?
It's in Hidalgo, Pachuca, Hidalgo. The university is more or less 100 kilometers from Mexico City to the north, in the middle area of Mexico City. Well, actually, now Mexico City is growing, and it's a metropolitan area, but we are in the border of the metropolitan area. More or less from Mexico City, the downtown Mexico City to Pachuca is about 100 kilometers to the north.
Have to get any permission from state or national to collect specimens?
Yes, yes. In Mexico, we have an institution, a federal institution that we call Semarnat. And then we need to ask by permission to collect any kind of samples. Although, for example, in my case, that I work with marine fishes, in general, I go to the, as Marilu, go to the markets, the fishes, like any person at the supermarket. Well, when I need to document all of these samples and put the information at the collection, I need to get a number and I need to give the data from Semarnat. So yes, we need to get a permission from the government. I have an license for research collect.
For talking. What would you say is the biggest challenge for the woman in that woman in science?
Can you repeat again?
Yeah, sorry. What is for you the biggest challenge for women in science?
Teach the students because, well, after COVID, the students change, the behavior of the students change a lot. And right now, the majority of the students doesn't have enough interest in different kind of the subjects. Looks like they go to the university to sit on the chair and just listen. They don't want to share or they don't want to participate very easily right now. They are very quiet. So the most challenge at the moment is teaching. And this is for, at least for me, and the other, try to don't have enough busy work because I want more time for the research, of course. Yes.
I was wondering if do you have any kind of outreach programs from the university or from your lab? As in, taking the science or this entire process or methodology to non-university students or high schools or?
Well, actually at the university I teach in bachelor.
Yeah.
I teach parasitology and I teach invertebrates. But also we have a program for master. So some of the students that they are at the lab, they are part of our lab. All of them are working with us. And I teach them how to fix, how to collect it, how to process different kinds of samples for doing some research. And of course, after we talk with them a lot about that, the importance about that. Sometimes I do, but it's well, yes, I do, but it's not very easy too because sometimes we don't have enough time. But the university, at least I know that the university and CONACYT is something like NSF, that they have kind of the science week, and we, as a professor or the resource person, we go to the school and show how they are the parasites and talk about the parasites. And now the government wants that as resource persons, we need to eat at all and teach, not just primary school or high school. They want that we go, for example, to the field and teach persons, the fishermen, and talk to them and say, okay, we call social working.
Yeah, that would be my next question, if you also work with the fishermen.
Yeah, we do. We need to do actually.
Yeah.
Yeah, we do.
Great, well, thank you very much. So it's 10:15. You know where our schedule is? I don't know. Yeah, so. Do we need to have a break?
Next one's not scheduled
Till 11, okay, so it's
till 11.
10:15.
Break.
Supposed to have coffee, but Gabor, do you know about where the coffee is?
At the student union.
I think we might have to go to the union.
think they would break it up because we have a schedule.
Oh, yeah, we are a little ahead. Okay. So what should we do? Just take a break and look around the museum for a while and show back up here?
Let the online audience know. That
Yes.
We'll be back at 11.
Okay, so hopefully this is working. We will take a break now. We'll be back at 11 o'clock. Anybody who's watching can also take a break, go to the bathroom, take a break, and we'll be back here at 11 o'clock to hear Valeria Trivaloni give her talk. So thank you very much for being here, and we will convene here in about 45 minutes. Okay, so anybody has questions, please feel free to ask. The parasitology exhibit is right here, which is very convenient for us. And this big tapeworm, bunch of specimens to look at. Hanson Pritchard's birthday, which is today, the 16th of July. And she turns 100 today. Today, we're going to hear from Valeria Trivaloni, and she's going to speak on tracking the elusive, uncovering high diversity of plant pathogenic bacteria using museum specimens, which is really, really cool. This is the kind of stuff that we need to be using museums for. Thank you.
Thank you. Okay. First of all, I wish to say happy birthday to Marilu, of course. And I also want to say that if we are capable to do something different and contribute with novelties in science, for some parties because of what we inherited. So I'm really honored and proud to be here today to celebrate the legacy of a pioneering woman of museum-based science in parasitology. And even though I was not a student of Marilu, I've been certainly influenced by her accumulated knowledge through the training that I've been receiving from her former student, Dan Brooks. So I'm deeply grateful to be here today presenting some perspective of my research, which include part of this inheritance. So I don't know if I'm gonna live 100 years, but I will make sure to pass by this precious inheritance and some novelties, if I can, to the next generation of young person that want to contribute to museum-based science. This is all I got. So this will be the outline for today. So I'm going to talk a lot about biorepositories, use them to track some elusive biological association and parasites. And then I'm going to kind of talk about my study model, which is phytoplasma, a group of plant pathogen, insects and plant association, and then how to even use biorepository to assess the risk of outbreak, and then some conclusions. So first of all, according with the National Cancer Institute, a biorepository is a facility that collects, catalogs, and stores samples of biological material, such as blood, tissue, salt, DNA, RNA, proteins, from human, animal, plants, for laboratory research. And of course, museum biorepository assure the long-lasting preservation of all these things. So, but then collection data archived in museum. So we know from many sources, and probably this is one of the first that have been published in 2004. We know that are under threat for way too long right now. And this is due to budget cuts and other institutional pressures. And these things, unfortunately, leading to collection being neglected, damaged, lost forever, and unfortunately, erasing centuries of work and countless of biological specimens. We have some example here in Lisbon the fire in 1978, and then unfortunately in Rio de Janeiro in 2018, and in Nova Delhi in 2016, and again in Brazil, this little town, Belo Horizonte, 2020. This is a little museum that preserved probably the biggest herbarium in Brazil, and was lost because of the fire. And still these days, there are many examples, but I just mentioned a few of them, like the Plata Natural History Museum in Argentina is facing a lot of problem during the budget cuts due to some government decision that cut till 20% of the public workforce, and cuts at the culture in general. So museum biorepository and archives preserve material and data for projects in several research fields. So now I'm going to just provide to you some example about what we can do with this biorepositories and informations. So in some fields, like for example, biodiversity, people are using data to estimate global biodiversity loss. Here, just an example. In this paper in 2018, they show that when museum vouchers are available and information is digitized, which is crucial part, we can actually evaluate the diversity decline over time. And in this picture, you can see here what we have right now in terms of species richness of bees, syrphid, and butterfly. There are 3 groups of insects. And you can see that where the countries are white means that there are museums, but the data are not mobilized, are not available. So we cannot use them. So just for a few colored country, we can use actually the data to estimate the change in biodiversity through time. So I think this picture is pretty indicative that we need to mobilize the data. Using museum biorepository for studies such as in ecology, people have been using this data to reconstruct the human impact, especially across Anthropocene. There is still a debate when Anthropocene had to be like the date where Anthropocene starts. Someone says 1974, someone wants to set this date 1994, but the reality is that we have this pressure amount of data. And in this paper in 1918, Andrew et al. Showed that despite some sampling for bias, human population level have a positive impact on biodiversity of epiphytic fungi. And as you can see in this picture, population density and diversity of epiphytic fungi in an herbarium museum is increased. So phylogenetics. Many people have been using data to revise phylogenies and taxonomic status. This is a pretty old paper. There are many more that are more recent, but just like what they did with the finches. So these two guys in 2002 reported several errors in original identification, and they went through the collection again, and they were able to use all their museum specimens and provide more data about taxonomies and phylogeny, also using taxa, extracting DNA from taxa that are even rare or extinct. Evolutionary biology. Here we have a paper from Klimov et al. In 2007, and they observed this reverse codivergence, or if you want to call incongruence, even though they wanted to prove some kind of co-phylogeny and coevolution between a group of long-tongued bees and their parasite mites. So this was pretty cool because they went through more than 300 specimens, voucher specimens, and they were able to retrieve mites, and they were able to run DNA, so to extract DNA, to run a phylogenetic tree, and they were able to study the cophylogeny, and in this inferred speciation within, they were able to infer this speciation within hosts, and now switching that apparently was the most parsimonious processes. As you can see here, some early derived mites are associated to ancient lineages of bees. So they were expecting something else, but actually using this museum biorepository, they were able to rebuild the evolutionary history between the association, which is another thing that we are trying to do with our biological model. Population genetics. These people in 2019, again, conducted several studies on museum preserved cockatoo, and they were like studying several specimens from all over Australia, and they concluded, because they wanted to prove that using material from these specimens is possible to gather reliable genome-wide SNP data that allow us to distinguish between population of this bird, and they successful retrieve this kind of genetic data from museum specimens that were, by the way, pretty old. So another group of study in toxicology, they try to usually reconstruct pattern of pollutants exposure. This is a nice paper, recent paper in 2023, and they basically define the standard for preserving fish specimens that will be suitable for tracking contaminants, such as, for example, mercury over time. They actually pointed out that if you preserve the fishes, I'm not an expert in preserving fishes, but they published that if you preserve fishes in a certain way, you will be able to measure the mercury that come from exposure of the fish when it was alive, and not like past contamination of mercury. So you are able actually to track the contaminant through time, which is another amazing thing. Again, environmental monitoring. This is another interesting paper. I like that a lot, on a field, this group of bird, the field sparrows. So these two people in 2017, they wanted to track the atmospheric black carbon between 1880 and 2015 within this US manufacturing belt. And basically, as you can see from this picture, the black carbon peaked during the first decade of the 20th century. And then it was positively correlated with coal consumption until 1970, when there is this clear air legislation that caused a decoupling within with the black carbon declining as the consumption of coal continued to rise, actually. And you can see here the consumption rise, and actually the black carbon decreased because of the effect of this legislation in the United States. So, and all of this analysis were possible because they were able to retrieve the black carbon from the feathers of the bird preserved in the museum biorepository. Pretty cool. So here we are, pathology. So for parasitology in general, the use of museum for such application is relatively recent. Of course, for me, as for many people here, Biorepository provide tools to answer fundamental questions about biological association, so test theories and hypotheses, but also critical evidence for solving problems that science, but most importantly, society are facing. As Brooks says long time ago, parasitologists should deposit voucher specimens of all host species examined in the course of survey or inventory studies, including purely ecological fit studies. This will help a lot, but actually rarely happen. So we know that when we document host pathogen association, this preservation of host voucher specimens, so both the host and the pathogen, is critical. But this practice of vouchering is mainly performed by taxonomists, so they just obviously focus on the taxa of interest, and they meant to run morphological study, and other subdisciplines often overlooked the importance of vouchering. So disciplines as, for example, genomics and epidemiology. And in this paper, actually, you can see that when usually people want to extract DNA from a voucher or a specimen, they overlook to voucher the individual. And this is what they found, like running a literature review, so that just a few portion of DNA sample is actually vouchered. All the rest in dark blue is not vouchered. And this is for many group of vertebrates. So in particular, this is my focus, I would say, for vector-borne pathogens. There is in general no mention about vouchering the host, whatever it is the vector or final host. There are few exceptions, of course. So I just would like to present this because I've been traveling the last month for vacation in Italy. And it happened that I was looking at, I was in Catania, and there was an exposition for Caravaggio. And there were many paintings from other people that were contemporary, that were that period. Caravaggio was an interesting guy. And there is this one painter, which is Fede Galizia, that happened to be a woman, but at that time, painters in 50, 78, women painters were mainly unmarried, of course, and none. So this is say a lot, so they are a lot of time, and they were also about crazy about okay, this is in Italian, unfortunately, natura morta con studio di pere, which means that nature studying peer. And what happened here, that I immediately noticed this, I mean, with my parasitology eye, said, cool. This is actually what we call Venturia pirina. So the disease is the black spot of pear, is a fungi, and the genus is Venturia, and was first described in 1882. But because of the painting, we can date back the presence of this disease at least to 200 years before, which is kind of amazing. And this is not just because me going to see the exposition of Caravaggio, this thing of looking into the piece of art and for taking evidence of plant pathogens is something that people do continuously, but actually is, yeah, an interesting perspective if you think about preservation of specimens. We cannot come back to the physical specimens here, but at least we have some clue about the temporal progression. So From the host pathogen association point of view, cataloged records and voucher specimens are the primary source for tracking past host pathogen association and infer evolutionary pathways. This is what we do. Documenting association in no longer accessible collection sites, which is pretty convenient, revising the taxonomy status of the associates, and then resampling the association over time when new technology became available, such as new genomic approaches and stuff like that. So this is what I found lately about tracking of pathogen association using museum biorepositories. There is this group in 2002 that study antiviruses, and they know that they have this strong association with reservoir species, and they basically wanted to determine whether this new strain of virus, which is called Cynombre virus, if I pronounce correctly, they basically were hypothesizing if was historically present in this region, Central America, in 1993 or before, because was detected in 1993, but they wanted to know if actually was before. So what they did, they went through the blood of more than 700 cryogenetically preserved rodents, and they tested them, and actually they were able to find this new strain. So they went back the presence in the region, and there were also some recent screening in other museum tissue that actually detected new antiviruses of unknown human pathogenicity all around the world in several other host types, such as, for example, bats. So we also know that museum biorepository are really important to predict emerging infection diseases. And someone realized that, and actually in 2015, there was an interdisciplinary workshop on scientific collections and emerging diseases and infection, where people met and they tried to explore opportunity for networking and collaboration. And I would say that after that meeting, something has been done, mainly for zoonotic diseases. We have few examples. For example, this paper, basically they have this program where they used two museums to kind of routinely analyze bat blood sample with the purpose of pathogen surveillance. So they have been doing many work on that, especially in this museum where the specimens and the tissues are well preserved. There was also a group of people that in 2017 tried to publish like kind of workflow for everyone, where they tried to depict the museum approach to track pathogens, and they put the Natural History Museum in the center, and they tried to explain how the information can flow within and out from the museum, and many people can take advantage, researchers, stakeholders, public, citizen science, and so on. And of course, as I said, they were still thinking about zoonotic diseases. Yes, eventually there were another paper from Colella et al. 2020. They depict with this picture how many information from a single voucher you can get. So genomic data, of course, parts of the specimens, which is tissues, blood, DNA, whatever, but most importantly, field notes. This information are going to be really useful in the future, and we have to take care of digitized using standards, which is another huge thing, and everyone have to agree upon. And every single detail had to be available for everyone. So eventually you have also associates, which is another, I would say, specimens that came from the museum preserved voucher host. So an interesting paper just this year was published. Finally, people that used to study malaria realized, malaria epidemiology in particular, realized that probably it's time to go back to Culicidae collection. So Culicidae is the family of a mosquito that is most important in spreading different kind of parasites, including viruses. But unfortunately, still in this paper, they are prospecting the use, they are pointing out how important it is, but still there is so much work to do. And this is just zoonotic diseases. For plant diseases, this is the thing. For plant diseases, museum specimens have been overlooked. Unfortunately, we have just one example in 2017 where they are studying a disease that is called zebra chip disease, usually affecting potato, other crops. And the vector of this disease is, which is a kind of Candidatus, it's a bacteria, Candidatus bacteria. And the vector is this specimens of psyllid, which is which is an Amiteran insect. But unfortunately, they were not using physical specimens. They just, which is something, they used data, the occurrence of the vector in this area in California, and they were able to predict the spreading, like what is the probability to have a certain population of the vector through times, but this is all we got. So unfortunately, we cannot track the pathogen itself using just the data. So we have to look into the specimens. This is what we actually are doing. We published with our group at University of Illinois, a paper in 2021, where we used the Illinois Natural History Survey insect collection. And it was just for fun. Let's see what there is in terms of pathogen inside the specimens. And we first discovered that there's a group of plant pathogens called phytoplasmas. And they were present in relatively a lot of specimens worldwide. So, and then with the group that we share idea with Scott, Dan, Walter, and Eric, and other people, we have been publishing how important it is to preserve and use a museum biorepository to document this association. And in this paper, we were able to point out that it's not just all about zoonotic diseases. We must include the plant diseases, but most importantly, we need to treat them as the same case of study because they basically work the same, and we have to approach the problem in the same ways. So documenting our model study, which is phytoplasma, which is a group of bacteria that is pathogenic for plants and more or less commensal for the vector, which is an insect. We know that this museum-based science is really important for several reasons. So we know that disease from this bacteria is becoming a huge concern. We have a lot of emerging phytoplasma diseases worldwide. These diseases are crippling entire industry. There are many infections in all over the world. So, but we still don't know nothing about vectors. We have few data about vectors. And also we know some region we know very well, some other regions are neglected, overlooked. We have a lot of problem from taxonomical point of view. And of course, all of this biases limit the inference on potential, on predicting potential emerging diseases, phytoplasma diseases. And here we come, the biorepository and archives may provide this baseline to study these associations. So starting from our insect collection at Illinois Natural History Survey, this is an overview of what we have in terms of insects. So we have over 1 million collection of objects, and among them, different groups. My group focus on Emictera auchenorhyncha insects, which is this one. They are important as vector because as for mosquito, they fed on the phloem or the xylem of plants. So they are subfeeders. So this is how they can acquire and transmit pathogens from plant to plant. So in Natural History Survey, we have over 144,000 dry specimens of this group of insects and more than 500,000 ethanol preserved specimens. So this is when 2 years ago, we decided to apply for a National Science Foundation. And thanks to this project, we are pursuing, as the name of the project says, 2 goals. So first of all, we want to discover the true diversity of phytoplasmas. And second, we want to assess the potential for emerging phytoplasma diseases. So about this first part, so as I said, vector-borne diseases, this is my model here. There are 3 things. So what we call usually the final host, then there is the pathogen, in this case it's phytoplasma, and then there is the vector. So first of all, we approach this problem like studying the vector and the pathogen association. To do so, we got this 500,000 samples that Chris Dietrich, which is depicted here, have been collecting for more than 20 years at Illinois Natural History Survey and have been preserving properly. So this is why we now are able to screen the specimen and extract the DNA for the presence of phytoplasma. Yes.
Are there, he has frozen specimens for a lot of them?
Yeah, frozen, ethanol, 96%. So really, we have a huge amount of material that is properly preserved, ready to be used, and digitized, finally. And most importantly, worldwide. So I had a lot of fun to take all the georeferenced point where Chris collected for over 20 years into this map. And I plugged the points on this raster because I wanted just to show his focus. So the white region are croplands, black are natural area. So almost all the points fell into natural area because Chris is the taxonomist and systematic person, reference person for auchenorhyncha worldwide, and they have been interested in uncovering the diversity of this group of Emictera insects, and he was mainly interested in collecting natural area, mainly. Sometimes also in the interfaces with cropland, but mainly in natural area. So, and this collection were made between 1998 till today, okay? So we screened 2,000 specimens as a preliminary study, and this is what we got. So I put a phylogenetic tree here, in black, the strain that are known, in red, the strain that we screen in museum biorepositories. We got a lot of novelties here. So first of all, we detected 11 unknown strain of phytoplasma, never detected before. So in natural area, in specimens of vector that were even undescribed. So it's amazing. I went to the collection from Chris. He didn't describe the species yet. I screamed. I found a new species of phytoplasma, and this time is a new species of phytoplasma inside a new species of lithopter. So it's a double discovery. It's amazing. And then we documented new association, because you know there is a lot of work about having this pathogen always associated with a certain group of vectors and blah blah blah. But we are discovering a lot of new association here. And then inferring association with plant hosts. Why? Because I'm not going to present this here today, but what we are using an next-gen approach to detect the most informative loci for phytoplasma, but we are also targeting DNA from plants, which means that we can this time determine which phytoplasmas are inside the insect body, but also where the insect fed on, because we can determine DNA from plants in the gut content of insects. Kind of closing that triangle that I showed you before, where all the vector-borne pathogens are based on. So we have this complex epidemiological situation. So back again to our triangle, I have here some news about the plant pathogen association, because as I said, we can analyze the vector, we can get data, DNA data from the plant, from the pathogen, but we can also analyze voucher plant from herbarium. So I didn't, actually, this is not my idea. I didn't thought that was even possible until when, almost 3 years ago, this amazing person, Alison Arrington, I didn't put her picture because I didn't have time to get her consensus, but she's working in Michigan Herbarium collection, University of Michigan Herbarium, and she's nothing. She's not never involved in plant pathology or pathology. She's a botanist. She's a taxonomy, an amazing curator, but she went through this archives and she came up with this idea. She was studying voucher specimens, and I just quote her words. So peach yellows, which is a disease that was observed in 1880. So peach yellow, Sanso Smith, is indeed the same disease that was later, years later, like maybe more than 60 years later, called X disease in the United States, and recognized to be caused by phytoplasma just 60 years later, despite the fact that the Smith's work does not appear to have made into the contemporary literature on pleural infected phytoplasma. So despite this, eventually, we need to look into this pressure material. So she immediately realized that if we can get DNA from these vouchers, we can track back the identity of a disease that is causing a lot of problem in the United States right now. This is the disease. The X disease of stone fruits mainly is causing a lot of losses. Just from 2050 to 2020, United States is losing over $65 million dollars annually. And this is how the symptoms looks like. It's really bad. And this is what they are observing. This is a recent paper from my colleagues in the state of Washington, and they have been observing outbreaks from 2017 to 2022. And now the situation is getting worse. So this is what we are doing. So Allison, we're collecting this data. This is a specimen from University of Michigan Museum. This is Prunus, was collected in 1887. It's symptomatic. You can see here on the left, the symptom is like elongation of the main stem and few grouped leaves on the apex. And this is the healthy plant. Looks pretty different. So Smith at that time were able to recognize this as a symptom of something. He didn't know what it was. But eventually he wrote down that was something, probably diseased. And then here in Maryland, same age, and the notes quote, I quote here, says, trees stunted and unhealthy, but probably not incipient yellows. So they've been noticing these problems and they've been keep tracking of this. And then I have another amazing example. In the same herbarium, we have Trillium grandiflorum, which is a plant that grows in the understory of forest. This is pretty amazing plant, and the healthy plants look like this. The diseased plant look like this. So pretty nice plant as well, even though it's disease. So it's a highly distinctive symptoms of infection, which are not, strangely enough, are not associated with any kind of decline. It's just a change of phenotype. And you have to notice that when it comes to prunus, which is a crop, we have these strong symptoms that bring the plant to die eventually. But in nature, more often plants cope with the pathogen. They just change the phenotype eventually, but they rarely die. So probably the pathogen also don't spread so aggressively. So in the herbarium, we have a specimen of Trillium from 1988 and 58, and botanists have been thinking that this is a new variety or maybe a regional form. And if you can read it, basically they got non-infected on the left and on the middle, and this is infected ones. And the difference is there's coloration on the middle of the petals and the petiole, which is longer in the infected one. So botanists just start to describe new forms of plants, like thinking, oh, it must be something different. So here, Trillium, new form, Forma striatum. So they've just been documenting this stuff like this. And then later, in 1996, and this is not by chance, when actually the plant parasitology people were able to determine, because in the meanwhile, DNA were, PCR were invented, so people that become to observe more carefully bacteria in the plants and viruses. So they defined that actually they discovered phytoplasma, but they didn't know that was another group, and they says, oh, they are mycoplasma-like, like mycoplasma that infect vertebrates. So it seems like mycoplasma, so mycoplasma-like organisms. And in that time, these people eventually heard about it. So in their volume, in the notes, they start to write down probably mycoplasma infected. So someone raised their attention about it. So it's just in 2016 that we were able to actually detect the phytoplasma that is associated with this natural plant, this white plant Trillium, which is related to the same strain that affect prunus in the United States. So again, there is this link between wild area and crops. This is not a surprise. This happened with zoonotic diseases as well. So, and here what we did, I'm actually processing the data right now. We were able to successfully extract the DNA from specimens from 1993 to 1996, and we created this temporal series. So we analyzed 26 samples of herbarium. We were able to retrieve the data, the DNA. I have the data. I was just downloading this morning. So this is all I got for today. So what about assessing the bioadsorbs, so the emerging diseases? As I said before, we can use specimens, we can use data. So this is what I did actually. I'm going just to tell you that for phytoplasma diseases, traditional studies focus on crop. As for zoonotic diseases that you want to focus on human being as the most important species in the world, for agriculturists, you want to focus on crop. But actually the answer is in the wild. So we have to consider both. We have to consider the interfaces between system, agro-ecosystem, natural area, and so on. So because of this bias, almost all vectors are documented in agro-ecosystem. We are missing a lot here. And we also know that only 10% of previously documented association involve non-cultured plants, which is a huge bias again. So this is why we decided to take these specimens from natural area and focus on natural area to balance a little bit this bias. And we have been published this diagram to depict the importance of having all the habitats into this conversation about pathogen, between pathogen and hosts, including vector, because where the things happen is where the habitats overlap. So we know that we have to answer some question. We need to know if pathogens that are spreading around are known or unknown. We need to know where they come from. So it's mandatory to study every single habitat. So what I did, actually, I use a machine learning approach, and I selected important variables that are known to be driver for pathogen and organisms in general for spreading around. So I got anthropogenic and climatic variables, and I modeled, I built a model to predict the occurrence and the outbreak of phytoplasma worldwide. So now the question was, we have data from the agricultural world on the left, and we have data from the natural area on the right. So we need to treat them carefully. So initially we decided to run two models independently, right? So this was a work from my undergraduate student that was doing this program. And then later I analyzed data from natural areas. So this is the comparison. So basically the take home message here is that we have a few of data that are very important in driving the presence of phytoplasmas, the pathogen in a certain area, in certain vector. And if you compare agroecosystem and natural area are almost the same. So population density, temperature, the amount of cropland in the surrounding of the point you collect, and precipitation. For natural area, we have another variable, which is the an index of environmental like a performance, if you want. It's a global index. So how well is doing that ecosystem. And for agroecosystem, we got the altitude. But for the other basically variable, they are the same. So here are the results. This is a decision tree. So why we like to have this decision tree is because, so after building the model, when you go out now and you got more data, and now you want to know what is the probability to have occurrence of pathogen in that area, in that condition, you can do it with this decision tree. So it seems complicated, but basically you have on the bottom, the leaf tell you the probability that there is an occurrence and what is the probability. And now you go through the tree as according which variable you are dealing with, you got a choice or another. For example, if in your sites the precipitation is less of 260, blah, 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 yes, you go left. If no, you go right. So you answer the question for your sites, you arrive at the end and you know the probability that you have to have an outbreak in that spot. So this is pretty powerful tool for assessing emerging diseases. So now I'm going to conclude. So we know that food insecurity is expected to increase in all countries. So including the countries that now are considered secure. And this is because we have two things. The first is environmental changes in general, including climate change and diseases. And unfortunately, environmental changes affect directly food security, but also diseases. So diseases affect directly and indirectly increased by environmental changes on food security. So it's actually getting worse. We have several indicators for food security that have been published lately, as you can see here. And for many of these countries, especially Africa, Latin America, we are going to experience severe food insecurity. One of the indicator, as you know, is the price of commodities. And I just wanted to bring a couple of examples, one from the world of crops and agriculture. So we have been observing lately an increase of cocoa prices that actually it is records about after like April this year, and then kind of decrease, but still pretty high with respect to the end of last year, as you can see here. And then in blue, the percentage are the GDP in the United States, and you can see how this is decreasing as well. And we can correlate this with one disease, which is affecting badly the main exporter of this cocoa in Africa. Many countries rely on the production in Africa, and lately they are experiencing troubles with this virus that is vector. The vector is actually this group of Eniptera, and it's going pretty bad. But we have also, on the top of it, another disease which is caused by fungus, which is fungal black pod disease. And altogether, this problem has been getting worse with climate change. So this is what's happening. And for another example from livestock, this is the bird flu in the United States. And as you can see, and as you probably know, the price of eggs have been increasing, like doubled since last year. And again, this is due to climate change. And actually, the bird flu, some variants right now have been even detected in human beings. This is not surprising again, but still affecting food security. So overall, this is like a summary we published lately. There are many direct and indirect economic impact due to the most important emerging plant and livestock diseases. These two things actually work together, impacting the food security of countries worldwide. And this is just a snapshot. I just wanted to show in temporarily we have like spike and losses in different countries for plant diseases and livestock. And this is another paper. We presented the graph of the cost of a new emerging diseases, as you can see here. And you can see also that after 20 years, the cost is internalized as a fixed cost that became cumulative. And on the bottom, you can see that you have this trend for the next 70 years, where this cumulative costs of emerging diseases, both in crop and livestock in the United States, will increase the food insecurity because eventually this curve exceeds the curve of the predicted GDP for in the United States in 70 years from now. So this is all I got for today, and thank you.
Any questions or comments?
In your decision tree analysis, do you use those pathogens and plant insect, you use them all for the input, or you select?
No, I used just the host insects association by knowing that in agroecosystem, when you have a documented insect pathogen association, you have for sure an outbreak. So no one is going to document an association just because sake of knowledge. Just because there is an outbreak. So that decision tree, this is why I kept separated. I'm still thinking, figure it out, but it's good to compare for right now. So for agroecosystem is associated to outbreak. For natural area, it's completely another story. We are exploring the association between pathogen and insects, so I can put this in a decision tree and predict the occurrence. So it's nothing related with the outbreak because we don't have any data about pathogenicity, virulence. We are actually kind of figuring this out. We are doing cool things like kind of quantifying the replication of pathogen inside insect voucher. So we are about to publish this, which is an indication of how well the pathogen is doing inside individual specimens. But we are going into some trouble with population and stuff like that, but we can do that. But for natural area, we have a lot of problems. So we are not doing plant pathogen, except for this collaboration that I have with the University of Michigan, where I start to screen for pathogen. But for right now, we are focusing just on this strain because we have such amazing notes from their barium. So we just focus on that strain. We are expecting the strain, but this is just an endless field of research. I mean, there are many things, but for right now, just focusing on strictly on insects and plant in terms of machine learning, insects and pathogens.
So I was wondering, is it among your plants, or is it possible to like take the machine learning algorithm and then run it to identify transmissibility between separate clades and strains? and whether or not all of these variables can have a differential effect on different strains of the phytoplasma.
I think that machine learning is not the best approach. The individual-based model would be the approach there. Yeah,
But then it is possible.
yeah,
Right?
sure. Yeah, this is actually what we are trying to
And is
do.
it reasonable to find these kinds of differences where you have separate, like among the new variants you had identified and in the different clades, that some would be more or less diverse,
Yeah.
transmissible, different phenotypes manifested, things like that?
Yeah, this is what actually we are doing with a master student in Brazil. We are trying to take this into this simulation based on individual-based model where we parameterized several things that basically pertain to the pathogen as pathogen of both. So I don't like the word pathogen here, but to the consumer, let's say, but let's say pathogen, but that is pathogen of both. The vector is hosted by both vector and plant. And then we also parameterized the vector as the pathogen of the plant. So, and this model may take into account what you are, I hope that it's going to take into account what you are trying to ask, which is a very actually important thing to do, but we will see if we will succeed.
Kevin?
Thank you for your talk. It's great.
Thank you.
Quick question, I think I missed it. There was some infections turning big petal colors into green color,
Yeah,
and you say there is more attractive.
yeah.
Is it more attractive for the pollinators?
No, for the people.
For the, oh,
Yeah,
yeah.
the botanists just like it a lot, right? Oh, it must be something special, must be another species, a form, blah, blah, blah. So raising a lot of questions, and it's a charismatic plant for many countries, like, I don't know, Michigan, where this plant is widespread. So it's really, you see, did you see the picture of the forest with this? Like, it seems a fairy tale, like this understory, just amazing plants. So this is why people look a lot on it.
Anything else? Anything from the World Wide Web there, Gabor?
No, there is nothing.
Okay. All right, well, thank you very much.
Thank you.
That was really cool. All right, so for those of you who have not met Orshi yet, she's here, and she came all the way from Vienna.
Yeah.
And so she got in late, but she made it, so it's pretty exciting. We all set. We just need to find the,
Yeah, I need to,
so let's go ahead and put this down.
this is the presenters.
Yeah, let's go
Here.
to here. Let me kill this first.
Okay.
Let me kill this. Then we'll go back here to the web.
Yeah, no, this one, the Chrome, I think.
Oh,
that's
we
where
got
I was.
Chrome money?
Yeah, there. No.
Chrome's here,
Uh-huh.
and you have it here.
I have it there, and then just press play. Mhm.
Now we have to share the screen.
It's, yeah, it's shared, so I
Oh, we
think,
already shared, Gabor?
Three dots.
but it's shared, right?
Not yet, actually. I'm just
Okay.
sharing. Three dots.
Isn't it this one? the
Oh,
shared?
actually,
That's right there.
And then just click on the share.
Oh, yes.
And then select the present screen,
Um,
the application windows,
this one.
This one here.
Mhm.
share.
Should be good.
Yeah,
Then
and
we'll
then
go here.
just, press present.
You can maximize.
Just,
Yeah.
Yep.
Should be it.
And then you have to go back to, no.
This should be it.
I think so.
Does that
Can you
work?
check on your end?
Yeah.
Yeah.
Because it's
Works, right? Okay.
Okay.
All right, so hi, everybody. My name is Orsi. And I, first of all, would like to thank everybody for inviting me here. I did not know Mary Lou, unfortunately, and as I hear, it was quite the miss. But the person I do know is Dan Brooks, and I've been working with Dan for quite some time. And I think he's made a major impact on the way I look at research and the way I challenge things that I thought were unchallengeable and don't challenge things that should not be challenged, I guess. And so I think in terms of the effects that it had on my work, I need to say I have never worked in a museum ever. I work with viruses that are probably less appealing behind glass than like macroscopic parasites would be. But what I do, I think, have in common with what Mary Lou had been doing is I work on pathogen diversity from a very different but very relevant perspective. I look at why we need to map this hidden diversity, also from a very different perspective. And I also try to deliver this to students and people who are not necessarily involved in the sciences. So I think speaking as one of the ripples that Mary Lou had made in this world, I can say happy birthday and thank you very much. So what I'm gonna talk to you about today is has a lot in common with Valeria's research. But I think I really have come at it from a reverse engineering perspective here of seeing a problem and then trying to work my way back, coming from the evolutionary sciences, but then ending up somewhere completely different. So I'm an evolutionary biologist by training, but I work at the Medical University of Vienna. And that leads to a lot of interesting conversations with people who have not necessarily been exposed. And so I'm going to start with this topic that I've just named Evo Demo, because it's evolution and epidemiology coming at each other from very different angles. And I like to start my talks with these two tables, those who have seen them or not seen them, right? So these are two rankings, essentially, which seem to be very, very close to one another, having the US, UK, and then the Netherlands in third or fourth places. And we would think that science is here being repeatable and it's confirming its own measures. So the one on the left here is from what's called the GHS index, or the Global Health Security Index, and the other one is from the Kaiser Foundation, right? So they both work on large databases. However, if I give you a little background on what these two are, so the one on the left, the GHS index, this is from 2019, right? This is the measurement of how well each of these countries are prepared for a potential pandemic. And over here, the US came in as best prepared, the UK came in as second best prepared, and the Netherlands came in as third best prepared, looking at a bunch of different things on how well they prevent, how well they detect, how they respond, what the health service system is like, what kind of norms are in place and observed in the country, and what are the pathogen risks in that particular country. The one on the right side is the countries most affected by excess deaths due to the coronavirus pandemic, where the US came in first, the UK came in second, and the Netherlands came in fourth. And the date between the two, so between, this is 2019 of October from when this GHS index was published, and that one is from 2020 November. So, sorry, I'll just move this little thing here so that we bother. So in this very kind of short 13 months, science turned out to be not so repeatable. And this is something that I thought from an epidemiological perspective could be considered a red flag, right? So we put a lot of effort into predicting something where the ranking we got right, but maybe in the wrong context. And so this was number one, but this unfortunately should not have been unanticipated. Because at the same time we were creating the GHS indexes, we were also measuring what we called emerging infectious diseases globally, right? And in 2004, there was a paper published which identified locations and strains of emerging infectious diseases all over the world, and provided not only a list of occurrences, but also provided guidelines as to how we should start tackling this problem and what we should be doing about it. And after 2004, they did kind of a follow-up to this identical study on how well we had been doing in the past 13 years. So that was in 2017. And this was the result they got. So the red ones were the ones that were still around, and the black ones were the ones that had emerged after the 2004 study. So long story short, it was again something that we weren't doing terribly well in. And again, this is 2017, right? This is 2 years before the GHS index identified that here is the best prepared place, right? So this is the best prepared place for a pandemic response, and 3 years before COVID. And so the question came up, what are we not seeing here? Both from an epidemiological perspective, but also from an evolutionary perspective, right? There is some mistalk over here. And so this is what I became very interested in, because when we look at pathogens, the way we understand their evolutionary patterns and dynamics to happen is influencing how we think about what they do. And this is going to have implications on epidemiology, because based on how we imagine these things to work is going to determine how we look for them and what we look for exactly when we want to sample human, animal, or crop pathogens, right? So we kind of try to predict where they would be, what they would be doing, and go out there and look for them. And so when it comes to the evolutionary side, I'm going to try and do a little bit of interpretation here, which is what I commonly do at the medical university. I talk evolution and then I try to translate it to epidemiology, and then vice versa, right? Because there's not a very good dictionary between these two languages, so I'm trying real hard, right? So from an evolutionary perspective, right, we can model these things. And this is a model interpreted from the Stockholm Paradigm, the book. But there are different ways that we can model and different model structures that we can follow. So let's say that you have an environment for the host, which is what the host represents, right? And the host is going to offer different kinds of variables for that pathogen, be that anything, be that protein structure, be that nutrients, be that something. And so these variables are, let's say, X and Y axis over here. And the host itself is going to represent ranges within these variables, right? So it's going to offer this range on X and this range on Y. And so the interface, or the overlapping area over here, is what's available to the pathogen. And so you have a pathogen population where the genetic capacities, some of them or a lot of them overlap with what's being offered by the environment, but there are also some that are, you would say, suboptimal for that environment. They're either neutral or they can't be utilized, they just hang around. And so here is one of the ideas. Let's say this is time 0 when a pathogen just got dropped onto a phytoplasmigen, just got dropped on a crop, or a coronavirus just got dropped on a market. And so according to one of the models that you can follow, there's then some sort of selection expressed by the host, which triggers specialization, loss of genetic diversity because of the suboptimal traits that are present over there. So you're going to have a smaller subset of that pathogen survive in that host in time 0. And then this is what's going on in the ancestral environment. So when that pathogen needs to move to a new environment, this is the new kind of condition it's going to be working from, right? This reduced diversity because of the specialization. And the only way that it would be able to enter a new range offered by a new host is through mutation, right? It's got to mutate itself into that new little square because the ranges have shifted. So this is what you would say is the mutation-dependent model or the host-parasite coevolution model, right? This is one idea of how the evolution of pathogens could work. Now, there is another idea here, and I'm going to tell you why it's important to contrast these two, right? So the basic idea or the starting point here is the same. I've just dropped something on this host population, right? But over here, the big difference is that the diversity does not become reduced. Because in order for this diversity to reduce, you really need very strong selection to be acted on that population from some shape or form. It's not impossible. You know, we've all been there, kills 99% of all known bacteria, right? That's strong selection, right? Because the 1% or the 1.1% that survives, we're going to name MRSA, and it's going to be coming out of hospitals, right? But it does happen. But in nature or in natural habitats, maybe not so much bleach used, right? Symbolically and literally. So what happens here is that you maintain, or the pathogen maintains this genetic diversity. And that is going to allow it to, upon encountering a new environment, it had all these little genetic traits there that were just hanging out. They're conservative. They don't bother anyone. There's no particular reason to lose them. But they do allow it to move into this new population of either a new species, naive hosts, newly vaccinated hosts, but let's not get into that. And then once it moved in there, there's going to be a subset that's going to start diversifying in that particular host, and you have a new set of diversity. But over here, the genetic diversity does not go through these uniform loss periods and then expand again and then lose again, right? And so this is the mutation independent model, or the Stockholm paradigm, where now there is no particular new trait required to move into this new population. Now, if you contrast these two, you can draw evolutionary predictions from them, but you can also draw epidemiological predictions from those. From an evolutionary perspective, right, I keep coming up with this diversity stuff, right? And you can measure diversity, and I'm going to now zoom into like viruses probably, but you can measure it in anything. You can measure interspecific diversity, which when it comes to biodiversity is usually an index of how well an ecosystem is doing, right? You can measure intraspecific diversity, like within the same species, how big is the diversity, right? If we're having, I don't know, 30 kakapos on an island, then it's probably not as fantastic as having 10,000 rats on that same island. Unfortunately, the rats are going to be doing better in this case, right? And then you can even look at, even in a virus, within strain diversity, right? You can look at the diversity of an Omicron strain for COVID and how well that would be predicted to move on into new populations. You can look at diversity in human papillomavirus genotypes, right? And you can start talking about like antigenic escape and like vaccine escape and whatnot. So in terms of diversity, right? In the mutation-dependent model, you would have a lower diversity that you would get from this pathogen population, and in the other one, you would get a higher diversity. However, this is what's going to influence how we think. But when we move on to epidemiology, it's going to be influencing what we look for. Because when we do epidemiological sampling, we sample the hosts for the pathogens. And in our case, we sample people. And when we sample people, in one case, right, there are going to be cases. And in this situation, if this is what the pathogen is following, right, then my human cases are going to be coming from a very close phylogenetic relationships, pathogen speaking, right? Speaking pathogen wise. In the other case, however, my cases are going to be spread out, right? And that's going to make a huge difference because from the diversity, I can make predictions as to what the transmission pattern is between people. And then suddenly, I'm not only looking at phylogenetic trees, I'm looking at communities. And in one case, in this case, right, what I can say is that there had been an introduction to the community, and then it was passed from individual to individual. And you can, if you follow the arrows, you can actually map that very nice monophyletic tree of the pathogen that you're supposed to be seeing. And in the other case, however, I am going to be seeing multiple introductions of multiple different strains, which are all represented in the community, right? And that's going to make a huge difference because in one case, right, this is usually a rare event. It's unpredictable because it's a chance encounter of a pathogen, but it's boiled down to a single or few introductions, which means it makes sense for me to go and look for the introduction, the source of this whole thing, right? Going back to this Dustin Hoffman film of like, oh my, let's find the one monkey who's responsible for this. In the other case, it's going to be more frequent. It is going to be predictable, but it's impossible to track back where those things came from because it's constant introductions and this massive transmission chain or network or pattern. And a lot of times there are a lot of resources being dedicated to finding the source where it's not going to make a difference. Because if I find the purple source, I have two more that I'm not unaware of, right? And that's going to even make funding decisions. And so this is really a big difference that evolution can make in terms of how we understand epidemiological networks to function. So this is kind of the Evo-Demo version of it, right? Now, if we understand this thing, right? Now we are looking at communities and we're looking at the pathogens in the communities and where they came from and what the transmission is, right? But when we start looking into this thing, we need to look at what kind of data we're working with in epidemiology. And I think this is also relevant for museums because in most of the cases, what we have, medically speaking, we can only wish to have what museums have and the way they collect their data. And I'm going to show you why this is an issue, right? So here's my community. And then my question is, where's my data coming from when I'm looking at pathogen networks, right? So one of the things that we commonly do is individual testing strategies. We've all been there, right? Sticking swabs up our noses and our mouths and sticking swabs in all sorts of different places, drawing blood, doing this, that. However, outside of COVID, when everybody was usually more health conscious than we are in our average life, these individual testing strategies are going to be very good because they're high resolution, but they also have grave shortcomings. They are restricted, first of all, by symptomatic infections. Outside of a pandemic, nobody's going to go to the doctor feeling completely healthy and say, oh, please draw blood from me or stick something up my nose or any other orifice just to make sure. Or maybe those few do, but unfortunately, it's not characteristic for people to do that, right? It's also going to be limited by access to healthcare. Not everybody has healthcare. Not everybody who has healthcare can afford to go to the doctor because they work different shifts, because they're sole providers of the family, because they don't speak the language, they don't know the system, right? So this assumes that they actually go into a healthcare so that they have access. And the third, not everybody cares. You know, a lot of the times when we get fever and we start swelling up and everything starts flowing, the doctor is going to say, and rightfully so, oh, it's some virus. And I'm not going to go to my GP for some virus. I'm going to sit it out. I'm going to sit home, or I'm going to pop a Tylenol, and I'm going to go to work, and I'm going to go to the school, and I'm going to go wherever. And voila, in 3 days, it's gone. Yeah, it's gone, but it had 72 hours to spread, right? And we're never going to see this one because they never made it to these healthcare facilities, right? So over here, of my sick population, I'm going to get a very small subset. right? This is going to be very good because it's high resolution of the people who actually end up in a healthcare facility. I know their social background, I know their demographics, I know their medical history, I know the manifestations, I know a lot of information, but it's a very low catchment, right? So another option that we can do, and this is what the lab does that I'm a member of at the medical university, we can opt for wastewater. We can analyze wastewater. And this has been an up and rising star during the pandemic. And it's great, really, because you have a very nice catchment area. But again, you get shortcomings. There's granularity in sampling. In Vienna, it's great to analyze wastewater because we have indoor plumbing, we have sewage facilities. We can track it to building level where the sewage is coming from. But that might not be the situation in all of the metropolitan areas in the world, or in the rural areas, or in the underdeveloped third world country areas where the disease is going to be there, but I don't have access to this kind of thing. It's limited by things that I catch out of the wastewater needed to be shed into the wastewater. And a lot of things are not shed into the wastewater. SARS-CoV-2 thankfully is. RSV, yeah, also a little bit, but a lot of different things aren't. And it's very low resolution for the socioeconomic background, right? Mostly, I don't know who contributed to it. So in wastewater, this is what I get. I get the viruses, but I don't get the people, right? And there have been some ideas of, well, why don't we track human DNA from wastewater? Don't. just don't, okay? It's just not the ethical direction we want to go into. And it's a little bit of Soil and Green vibes over there. And so this is the thing with wastewater, right? Is that it's very good catchment, very high catchment, but it's low resolution. And so between the two extremes that are available to us, this would actually be the optimal, right? This is what a museum would do, right? You would get a high catchment area from all over the world. You would get the voucher species of where this thing was, what the ecological parameters were. This, from a museum analogy, right, is you go to North America and you go to Southeast Asia and you take one sample each, and then that's pretty much it. And then the other thing is I just walk through the door and dump a bunch of dead mice here with no tags. And then I say goodbye, right? And we're trying to do something in the middle. So how do I get a representative sample of my community who are suffering from this issue? And so this is where, luckily, there is a framework we could use for this, right? Which has been brought up today, which is nested in evolutionary biology, but it has a lot to do with how we can look at sampling and collecting and really getting the best of both worlds in this case. So the DAMA protocol, right, has been talked about today. Valeria mentioned the documenting and the assessment phase, but just to run you through quickly what this thing is. So in terms of documenting, right, it's a 4-step protocol on how to really make the best use of these preventive tools that are in our hands for pathogens, right? So documenting microbes found outside of epidemiological sampling. Outside of epidemiological means people who are not sick or don't know they are because they have no symptoms, people who have or don't have access to healthcare, and people who did not show up to the doctor, right? Somehow reach out to those. Then once we manage to document something from there, we can assess the risk, right? The phylogenetic relationship, the closest known pathogens and these conservative traits between the pathogen that we had found and known pathogens, and see what the risk is of this being carried over to a new population. If it's a high risk, we can monitor the distribution of the current and the naive hosts and also describe the interfaces. And once we have those interfaces, we can develop intervention right there. And we can adapt it to local conditions because now we've mapped where this transfer can take place and collect this really good high resolution data on the ancestral population and the potentially new population, right? And so within this 4-step protocol, what I really became interested in, this is why I said it was reverse engineering, was the second A, right? Because the first 3, if you try to follow my crazy color coding here, those are mostly nested in some sort of scientific discipline, right? It would be cladistics, phylogenetics, taxonomy, evolution, ecology, but it's a lot of different scientific knowledge put together, and that's great. However, when you look at the last one, the second A, or the ACT, now you're working with people. And the people are not just your community that you're working with, or not just the people affected, but it's science representing itself with evolutionary predictions and modeling and whatnot. It is also public health, because you need to understand the policy environment, what's feasible, what's unfeasible, what we can and cannot do, pros and cons of the tools, like the legislative tools in our hands. And it's, again, it's the people, right? Just because policy thinks it's feasible does not mean it actually is, right? A lot of the times, or most of the times, when you're dealing with a particular outbreak, it is a very specific population with all kinds of different cultural, socioeconomic backgrounds, priorities, and it's the same policy that you need to enact over here, right? And so this became kind of a focus, because if we want high resolution sampling, we have to work with all of these people over here. And so here is where I spent 2 years looking for a solution. And I ended up with this thing called transdisciplinary science, which makes the hair on the back of a lot of people stand up, because they don't know what it is, but it sounds very ooky-mooky, esoteric, whatnot. It's actually super cool if you come to understand it. And it's widely used in agricultural sciences, in food sciences, because in those places, you can't avoid working with non-scientists, right? You can't avoid working with industry, with private organizations, with government stakeholders, with the farmers, God forbid, who produce the crops that we want to investigate. I think disease epidemiology is yet to make an appearance on this stage, right? But what transdisciplinary science means is integrating knowledge, different kinds of knowledge across academic and non-academic stakeholders, right? And the main message here is that there's not going to be someone who speaks the truth and the others accept it. Everybody needs to accept what the other one is saying. And this is very icky territory, right? So none of government people are not used to this. Scientists are not used to this. The locals don't want to deal with our BS. Right? And so having found or linked this framework, the Dhamma protocol, to maybe transdisciplinary approaches, I spent a little time developing the theoretical background to all of this. And I put this TD, or transdisciplinary theory that matters, because there was a director who said that the institute I was working at is going to follow this logo, or this motto, theory that matters. And I really liked it, because I felt like it's a thing of, you need to have an aim, you need to have an idea and a goal of why you're developing a theory, not only for the sake of developing it. And so when we looked at how we can implement transdisciplinary sciences into disease epidemiology, we looked at how healthcare interventions are designed, right? So if we follow the COVID logic, or the COVID narrative that we've all been parts of, there are global institutions, right? There's the CDC, there's the ECDC, there's WHO, and they're responsible for overseeing the global situation of epidemics, pandemics, infectious diseases in general. Global institutions are going to be collecting data from medical facilities, right? Patient data, mostly. How many people manifest similar symptoms, similar etiology, within a time frame, within a geographical area, to kind of identify if there is an outbreak, epidemic, pandemic, something going on. And they're also going to be collecting from research institutions, right? Biological background, virology, bacteriology, medical treatment, care, whatever it is. And based on this, they're going to be making regulations and policy recommendations to regional institutions, right? These would be governments. This is a very wide variety. It can go like the European Union, national governments, municipalities, local public health offices. So it can trickle down to like smaller, from a larger catchment to a smaller catchment. And then at one point, this policy in some shape or form, usually extremely distorted and kind of word of mouth, but makes it to the population. And the population is going to receive a message saying social distancing. And that's going to be understood in some way or not, followed or not, adapted or not, right? I took it off, but this would be like a very good place for the picture we all remember with people wearing masks and then a hole cut through the middle to ease breathing, right? Policy adaptation, right? Policy is wear a mask. I have a mask, just don't like breathing in it, so cut a hole, right? So this is kind of how things trickle down. And so there are really major perks to all of this, because the fact that it's centralized and top down makes it very efficient. So for instance, in COVID, January 20th was when the WHO announced that it was a public health emergency of international concern, right? And within 2 weeks, governments started shutting down. Now, globally, this is crazy efficient. I mean, this is high speed reaction right there. It allows for this rapid response, and it allows for accumulating ginormous amounts of data and expertise, right? So these are definitely perks of it, but there are downsides to this whole thing too. One of the downsides is there's no direct feedback from the subjects of the policy, right? This almost never trickles back. If there are feasibility issues, if there is non-compliance with the policies, nobody ever knows, right? In fact, the way that sometimes they're trying to figure out is, again, through the medical data, right? We enacted this policy, we keep on catching pathogens from sick people, and the pathogen diversity suggests they're not doing what they're supposed to be doing prevention-wise. Well, like, the fact that they're still sick might also suggest this without having sequenced those pathogens, right? So this is one of the issues here. And it's also very limited targeted sampling, right? So when I explained medical facilities and research institutions, we merge subsets of the population, or we overestimate the representative value of whatever samples we have, and we have limited geographical catchment at best, right? When we look at these pathogens. So when we developed this theory, it was very clear that we had to do it on 3 levels. We had to go global, those are different recommendations, regional against something different, and local against something different, right? Unfortunately, there's no one size fits all. So for the global, right? Global theories, what we did is we looked at the conceptual model of global health security approaches. We looked at global frameworks that work with either different kinds of pathogens or different desired outcomes. And we analyzed how these measures and frameworks could be best, not changed, adjusted to accommodate for maybe increased sampling, maybe larger amount of measurements taken, maybe new databases that the specimens could be entered into. And we suggested ways of implementing this evolutionarily based prevention, right? And so based on this, just to kind of include this preventive approach into existing global frameworks, we kind of came up with this so-called 3P model. It's the prevent, prepare, palliate, right? Because this is dividing global organizations based on their desired outcome and targets, right? There are those that are more focused on the treatment And when it comes to disease, this cannot be neglected. Diseases, infectious diseases, sometimes have chronic complications, sometimes have a very quickly progressing manifestation. And we just gave examples such as the Medicine Without Borders, the Médecins Sans Frontières, the Bill and Melinda Gates Foundation, where a lot of the times the focus is on delivering care to those who are affected, right? This is the baseline. This is where pretty much medicine historically arose from, as treating the sick. Then, kind of pushing the boundaries and pushing the horizons a little further, we went from this original aim of medicine to prepare. And the EcoHealth Alliance, One Health Initiative, these would be good examples of understanding that diseases are more than what we observe them to be. They're more than the sickness. They're more than the symptoms. They're actually parts of the ecosystem, and we can track them. And over here, the basic idea is we just follow from the human disease or the livestock disease or the crop disease, follow the pathogen into the ecosystem and map out the non-human, non-livestock, non-plant hosts, and kind of get a better understanding of where this network and how this network is structured. And to this, we added the PREVENT, which is essentially the DAMA protocol, where we flip this around. And the idea is not to follow from the disease or from the human population outward, is to start mapping outward before it makes it back, right? That's where we can essentially cut this infection timeline and avoid an outbreak or prevent an epidemic, right? So this is kind of the basics of this framework that we tried to adapt and tried to build. Again, it's not about wiping the table clear. It's about trying to make this merging as painless and smooth as possible. Policymakers don't like pain, so we're trying not to be a pain anywhere in their body. So this was the global, right? Then we went regional. And our main targets were, how do we develop these feedback systems? How do we inform the policy-making organizations about the policy subjects and the feasibility and the success of that policy directly and not through indirect data collected? And so with the regional frameworks, this was the basic stance. So we sat down and said, here's an emerging pathogen that's emerging in some sort of population. What are going to be the different factors or the different groups that have an impact on how this pathogen is going to be transmitted? Well, it's going to be the patients, surprise, surprise, or the people who are infected, because they're going to be the ones where the transmission happens, right? They're the transmission network. They're going to be distributing or exhibiting certain risk factors to this infection. And they're going to be within some sort of an infrastructure, healthcare infrastructure, information infrastructure, which is also going to have an effect on how this pathogen is being handled. Then there's going to be science, because science loves jumping on this, rightfully so. It's going to be molecular attributes, it's going to be treatment, vaccine, host range, a lot of different things mapped on this pathogen. Then there's also going to be industry, because there are going to be sectors who are affected by crop diseases, by COVID, needless to say, right? The bright blue skies that we got during COVID were because one of the sectors affected was the energy department, and they had to stop the manufacturing and everything shut down. It's going to be product development, vaccines. If we all remember the price of masks, where it started in the beginning of the COVID and then how it progressed afterwards. It's going to be market exploration. COVID didn't take long for it to be marketable, politicized. There's always an opportunity there. So industry and private sector is going to be interested. And then there's the governments, of course, because they have a public health capacity that they're more aware of than anybody else for scientifically speaking. They're going to have resources that they can choose to dedicate or not. And they're going to be aware of the policy environment that all of this is happening in, right? Because pathogens, they're not only in a biological, they're also in a policy environment, right? That affects them. And so we were looking for something that accommodates all of these things. And then what we found to be on the intersection of taking all of these different social structures into account was what's called these living labs or multi-actor platforms, right? So this is, again, within transdisciplinary sciences, it's a structure and a framework that's been present in agriculture and food sciences, even in chronic diseases, such as, if I just say cardiovascular diseases, diabetes, right? There is a huge rollover there that the industry is taking, the government's taking, science is taking. There's a lot of communication with the patients, the R&Ds and the patient feedbacks. In infectious diseases, not so much, right? COVID was kind of a time for everybody to scramble to develop something similar, but then the pandemic is, I guess, now over, as everybody's claiming, and then this all stopped. And it would be hugely important to implement this same framework into the infectious disease landscape. So this is for the regional, right? And so in here, the knowledge types that we are combining is science is going to provide the characteristics and the predictions regarding to that pathogen transmission. The government is going to be providing the policy expertise and the regulatory role. Citizens are going to be giving feasibility and local condition input, and the private actors are going to be the practical and industrial implementation. So it's everything has to do with this particular pathogen system that we're looking at. And then finally, we went local. And on the local level, we decided that what we needed to do over here is reach out to and involve affected communities in planning interaction, right? It can't just come from somewhere above their heads. It can't just be a message that's on TV or somewhere and then everybody is expected to follow it. This needs to be done with the people who need to do it. And what this means is that we need to gather the local knowledge, we need to raise awareness, and we need to involve them in designing and conducting the intervention, right? They need to have ownership and a say in what's going to happen to manage this interface. And so this is something that we've seen during COVID, right? Is that when it comes to the same emerging pathogen, you had the community oftentimes because there's so many different communities, and they had their community-based organizations, environmental protection groups, neighborhood associations who were trying to manage these things. You had these local educational institutions, schools, for instance, with information, local community colleges. And then on the other hand, you had science and authorities, where the knowledge institutions, the university research groups, private institutions were giving input on what this thing can do. And public actors were also delivering particular messages. But as you can see, these are really kind of their own circulation, right? It's just like being within the yellow and within the gray. There's not a lot of overlap. And this is something that we said needs to happen. There needs to be a very big mutual ground here where it allows for sharing in both directions knowledge, right? And so this is the framework, again, within the transdisciplinary sciences, is what the citizen science initiatives can do, where you work with the community, but the community also works with you. They teach you about things, they give you input, they allow you to better understand the system that you're trying to work with, and not only imagine it the way you imagine an evolutionary model where everyone is just a dot in this white n-dimensional space, right? And so this is also something that we kind of not only tried to design the regional and the local level, but we gave recommendations on how these can be implemented for livestock, human, and crop diseases, because these are all different policy environments, right? So this would be epidemiology over here. This would be agriculture and food sciences. This is a different part of agriculture and food sciences. And we kind of showed not only how to implement each on the regional and on the local level, but how these two can interact with one another. These are organic parts, because the citizens enrolled in your citizen science programs are going to be the ones who are then in the living lab representing the community, because they've known this, they know how your system works, you know how their system works, and they can give advice. And so, having come up with this theory that hopefully matters, we decided, all right, let's see if it actually does, right? Let's do some, instead of TD theory, let's do some TD action and just test it out really if this is meaningful or if it just writes a few papers. And so we started on the local ground. And on the local ground, in the Austrian Viennese landscape, we chose respiratory viruses as our primary targets. For two reasons. They are characterized by rapid spread in interconnected groups because they're spread through droplet or airborne infection, right? And they have been responsible for pretty large scale epidemics. Maybe COVID-19 would be a good example, but also within the Austrian landscape, we've been having regular RSV outbreaks, influenza outbreaks that are recurring each year. And it's been causing some national news level disturbances, particularly in parents or workforces related to kindergarten to middle school age children, right? I was also a citizen of this experiment because I also have a kid, and I can tell you that anything that gets them for 2 days is going to get you for 2 weeks. And there's that affects the GDP, right? My 2 weeks affect the GDP more than their 2 days of infection. So there is again this rippling effect of the community of this pathogen transmission. And exactly for this reason, for the community, we chose high school students and teachers to examine this on, because they are disproportionately more exposed. They are highly interconnected, spend most of their days in confined spaces. They're a public health priority, because their sicknesses can have long-term negative health outcomes for the kids, but short-term negative health outcomes for the parents and the people linked to them. This is an age group that is usually not severely affected, so the morbidity is much lower in this adolescent age group, but they are in direct contact with under 5s and over 55s, where the morbidity increases significantly, right? So there's a lot of societal effects that kind of radiate out from this group. They oftentimes can be characterized with low compliance with prevention, right? There's this, there have been these instances during COVID where they would switch masks because they liked one another's mask better. So they just interchanged them in classrooms, or everyone had their masks on, but they would change lunch boxes because, all right, I'll eat half of mine, you can eat the other half, and I'll eat. Right, so there's not a lot of understanding, sometimes not a lot of priority given to these prevention measures. And then another just as important thing here is to empower students through this scientific involvement, because they were also exposed to these top-down regulations of you need to social distance, you need to wear a mask, and at the same time, I'm sitting in a bench with another student. The bench is not going to get bigger, the chairs are not going to go anywhere. How do I social distance? I'm forced to social distance on a bus and then sit 6 hours next to this random stranger from the other side of the city, right? So I think there's a lot of this kind of conflict, which we wanted to resolve and really kind of deliver something to them where they get a better understanding of how this thing goes, and we get a better understanding of our own limitations. And so in this case, we came from 3 different areas as to what we could deliver from this study. From a public health perspective, it's addressing the resistance that had been sparked a lot of times by these outbreaks and the feasibility issues in this particular community. From a data collection perspective, it's high resolution, high catchment data that we can collect from them. And from an evolutionary perspective, it's the unknown diversity, which is never going to make it into the patient data. It's going to make it into wastewater, but I won't know because I don't know where it's coming from, right? So this is kind of where we kind of tried to bring these three together. And so what we enrolled in the project, or the people we enrolled in the project, from the science and the authorities part, the medical university is represented by two research groups. One of them is the top three. That's me in the middle. And then I work in the group of Andreas, and a colleague of mine, Anna, is also participating. We represent the molecular group. Igor, further down there, he is the social epidemiologist, right? Or epidemiologist group. We also work with an organization who specializes in public health and outreach, and another one who specializes in interactive teaching and STEAM and STEM educational methods. And so together, we kind of tried to cook up a project that would be of interest and invited high schools in the Viennese metropolitan area. And what we ended up with is we have 6 schools in our program. This is Vienna. This is what it looks like, only not teal. So within Vienna, we have 6 high schools that we are currently working with, which add up to 47 students and 8 teachers in our pilot program. And the students named the project the Luftwies Initiative. It has to do with Luft is air in German, and the Luftwies, they refer to themselves as the airborne pathogen surveillance people. Right, so this is an acronym that comes from the German word. It directly, or the German name, it directly translates to airborne particle and pathogen filter monitoring with high school students, right? This is what they came up with. And so this is a citizen science project, right? It's in Austria, the very first sequencing-based airborne pathogen monitoring with high school students, or with any citizens for that matter. Globally, it's the third, where we look directly at pathogens, not through vectors and not through vector distribution, but we sample directly with the community, from the community, and then we do molecular analysis. And so we try to deliver on 2 scientific fronts. One is the molecular epidemiology, right? Because from this perspective, our hypothesis is that the sequencing-based surveillance is going to complement our insights into viral evolution, because we look at the genetic diversity, the strain diversity, the viral species or viral type diversity within this community, and that allows us to build trees. However, and this kind of probably ties back a little bit to what Valeria was examining, it's not only the genetic factors that you can build trees with. You can also build trees with the social risk factors, right? Sorry, yeah, on the other side, the socioepidemiology. Because if you look at the social risk factors, they're also going to have a different power on how likely they are to expose someone to this disease. This can be socioeconomic status, it can be ethnicity, it can be background, travel history, awareness, education, religion, and I can keep going on. But you can build this tree based on what kind of factors you're sampling in the members of the community and how that coincides with infection, right? And so as for the sampling, the molecular samples we collect in 3 different ways. We put an air sampler in the schools. This is a device, it's an active sampler, so it creates an airflow and collects on a little substrate and a cartridge everything that it had sucked out of the air. We run it for 24 hours. This is handled exclusively by the students, so we're not even there. They're the ones who exchange the cartridges. They come up with where this should be put. We also deploy a very scientific trash can, because we ask the students to discard handkerchiefs into this trash can, because those are also samples we can then extract. And we also collect swab samples. This is from the door handles, the light switches, any surface that is somewhere around this area that people who blew their nose, sneezed, coughed as they were taught, would then hold or open or touch, right? So this is the molecular sampling. And then for the socioepidemiological sampling, we run 2 surveys that we co-develop with the students. One is addressing misconceptions and the prevalence thereof, together with the demographics in both cases. Misconceptions such as, if I don't have fever, I'm not infectious. If my sibling is sick, but I don't have symptoms, I'm not infectious. If I reduce my symptoms with acetaminophen, I stop being infectious. So these kinds of beliefs. But we also ask about feasibility issue, right? So during COVID, we all heard that you should ventilate the rooms. And that's a policy currently in action in Viennese high schools. Now, when it's February and it's -5 degrees outside, this policy runs into an issue because what the students don't want to be is cold. And they don't open the windows because they're cold and it's not being ventilated. And guess when respiratory diseases peak? They peak in cold weather, right? So there are these kinds of feasibility issues that we're trying to map. And then we also run a feedback interview on the project. So the students who participated in the project, we sit down with the semi-structured interviews and we ask them to give us feedback on how well they thought this was executed. Did they learn something? Did they feel engaged? But also a little bit of reflection on, did it change the way they look at science? Did it change the way they looked at themselves? Did it change the way other people perceived them? To kind of draw them into this relationship between science and society and how this can be worked on. And so once the molecular samples, just to give you a little bit of methods, the molecular samples we put through a detection and quantification pipeline. We run a PCR. We currently have 2 bacterial and 8 viral targets. Those that come out positive, they go into the sequencing pipeline. We now use capture hybridization, so it's a target enrichment method. We use this twist panel that enriches for, I think, 40 different respiratory pathogens. And then we sequence it with nanopore first. And then to validate those, we use the Illumina sequencing. The sequencing results are hot off the bench in a sense where I have them, but I got them last week, so I haven't been able to do anything, right? I've not called any sequences or annotated anything. It's just there, right? This is where we are. The PCR samples we finished 2 weeks ago. So that's why the sequencing was able to go there. And then we do the bioinformatical analysis, right? We identify the sequences, and then the plan is to do the phylogenetic sequencing or the phylogenetic analyses. And so this is kind of the methodology about the molecular, right? And the socioepidemiology. But again, I'm going to loop back into the transdisciplinary sciences because that, again, is a whole different scientific method here. So we use this framework to build up the citizen science network, essentially, right? We recruit the people based on invitation, right? We have info sessions, teacher input sessions. We then co-develop the study with them. So the teachers are there. They give us input on what's feasible, how this should be done, how the students should be contacted. They manage the school, they manage the curriculum. They give us ideas how we can input this into the curriculum. Then there's the co-action. This is where the sampling takes place. And then the dissemination is going to come later this year. But one of the important highlights here is that any scientific publication that's generated out of this study, the students are going to be co-authors on it, right? So we invite them as co-authors. We help them disseminate the results in their own schools. They run poster campaigns, they run whatnot. And just to give you an idea, so the recruitment and the co-development, this takes a year of doing. So we started doing this study in January of 2023, and we started sampling February of this year, right? So this is the amount of time it takes to build a network, and I'm going to show you the results of that network. But once it happens, right, these are just impressions of the study itself. These are students who are sampling in their own schools. So this is how they manage the device. This is the sampling station with the garbage bin in the back. You see the garbage bin there. They have these little posters that they print for awareness raising. This is Anna and this is Fanny, two of the students that have been doing this data collection. Sometimes in some of the schools, they suggested we put it in the cafeteria because this is where the students queue for meals in front of the device. In other schools, it was put into the biology lecture hall. It's right there in the back, right? Everybody's super happy to be breathing into that device, apparently. And then yet other schools, we put it in what's called the Pauseraum, which is like a place where students hang out or in break sessions, right? So over here, you can see it's right there. That's the tissue collection, and the students are just hanging out in front of it, right? So this is across the different high schools. And then this is the students' contribution to the project so far and what they got out of it. So in some schools, the students put a little notification on the website, and that's how they kind of raised awareness that everybody should just come and cough into this machine. In other schools, they designed posters, right? So they use the logo that we designed together, and they have these things like, we want your germs. This means cough for science, science for the people, by the people. So they have these mottos laying around, and they also give talks about it. So they design their own little presentations. They talk to it, to parents, to other teachers, to other students. So hopefully they're really getting their kick out of scientific talks or scientific presentation. And then just to give you a snapshot of what the preliminary results we have, this is the molecular data collected in the project. This is one school over here. Each school was monitored over 4 weeks, twice a week. So you have 8 different sampling days, right? And in each of the sampling days, there are 3 types of samples. This is air filter, swab, and the handkerchief samples. And the dots are representative, so it's a heat map. Oops, it's a heat map. If you look at this down here, which is corresponding to the concentration that we're measuring, the samples that you don't see are still not on this database because they were still being processed. And what you see is that we consistently find SARS-CoV-2, RSV. There's diversity between schools. There's diversity within schools. Seems to be like a seasonality pattern there. So this is really just something that we're getting ourselves into to analyze this statistically. This is the molecular epidemiological sampling. From the transdisciplinary framework, again, preliminary results. This is a mathematical visualization of the citizen science network that we had built. This here is me. And if you go radially outside, that is corresponding to time spent. So what this shows is that it was kind of founded by a research group. The purple ones are the researchers, and the researchers I had contacted. Then this is the Vienna Open Lab researcher that we are working with. They're the educational outreach. They have a database of schools. So the invitations went out through her about this project to the teachers, who are now represented in pink. And teachers were the ones who then distributed and invited students, right? And so this is the starting point all the way to the 1 year mark of the framework. And what I can say so far is that looking back on the data, we have an over 93% continued engagement rate in the 2nd year of the study. So those who started with us, almost everybody finished with us. So students are still engaged. They're writing essays and projects. They're waiting for the results. So it's a continuous work that we can do with them. And so just to kind of summarize all this madness that I call my research. So this is kind of where we started from, that I was looking to collect high resolution, high catchment samples of pathogens infecting communities. And for this, we tested citizen science as one of the tools of transdisciplinary research. And it looks like, based on our project, that the data collected within this transdisciplinary framework is suitable to, on the one hand, uncover, sorry, let me just move this little thing here, right? To uncover this hidden diversity or yet hidden diversity of pathogens that we can then use to figure out which of these two evolutionary models are at play when it comes to transmission, when it comes to diversity. And with these, we can improve epidemic prevention back to the community and get a more complete understanding of what's going on evolutionarily and what we should do epidemiologically. And so with that, I thank everybody for the attention. As you can see, it's a pretty unorthodox approach to research, but I do think it's a change that we desperately need to manage infectious diseases. So thank you again.
Any questions or comments? Anything online?
Stunned silence.
Really interesting. The thing I was wondering about is how does the social media or the computer interactivity play into the way these students are talking to each other? Besides the posters, besides other things, because you know, I have teenagers and they just go, stuff just goes zap. It's out there instantly. And so it's very
I mean,
interesting.
it's a boost if you use it the right way.
Yeah,
So one of the things that we were kind of struggling with in this, or not struggling, but we were kind of not coming up with good ideas, was how to keep close contact with the students. Because again, understand, all the sampling is done by the students in the schools, right? I'm not there. I go there, pick up the sample, give them the new cartridge, and I'm gone. They do it by themselves, but they need to be in the loop somehow. And so we went and asked the students and asked them, guys, what do you think? You know, what would be a good way of keeping in touch? You know, where all the information of the project is there, but you can also keep in touch. And the first thing they said, oh, Discord.
right, that's
And if you ask your teenagers, they're going to know what Discord is. So we developed a Discord server for the project with different channels that respond to the molecular epidemiological methods, the socio-epidemiological methods. Each school has their separate channels so they can discuss school-related issues, sampling-related issues. They can write direct chats to me. And from an ethical perspective, because again, we're working with minors and it's the medical research, or it's considered medical research. Discord also has this very nice function where you cannot create an account without parental consent under the age of 16. So we have that kind of covered. So we're not pulling in minors into this shady looking online chat room, right? Which I couldn't sell it this way, but it's a Discord server that they can just access.
Right, I use it to teach.
Yeah.
So I don't use Canvas, I use Discord.
Yeah, yeah, and everybody knows Discord because
Yeah,
they're
all the
hanging out there
students
to
say, "Oh, you use
game or,
that?"
yeah.
They're much more engaged in teaching.
Yeah.
If I use Canvas, they say, because it's so tough to use. Discord is so smooth.
Discord is very easy. Yeah,
So,
so that's, yeah, I
Good.
think if you get the right info from your people, then it's a
Yeah.
benefit.
And then WhatsApp when you get to South America.
Yeah, although I think, yeah, WhatsApp has,
Yes.
I wouldn't use it for this because you
Probably not,
can't
yeah.
post things in there
Yeah,
that they can then, find their ways through.
Right.
So you can't do separate channels.
right.
I think
Well,
Discord,
they're actually coming up with some now.
that would be
That's
nice.
new, Yeah.
Yeah, but Discord also has a mobile app. I
Yes,
mean,
use it.
one of the things that we're doing for the paper we're writing, or one of the papers, is that the network I just showed you. So we want to use the edges to represent, so the width of the edge would represent the intensity of communication and involvement in the study. And I actually have a student, as we're speaking, who is extracting the Discord chats from the server and looking at how many times we had been directly in conversation with student 1 to 47, and then to kind of manage the edges or the width of the edges to show how much the students are engaged.
Anything else?
Well, I wanted to ask you a question
Yeah.
back, but also in your reading, your work on COVID-19.
Yeah.
were there some, like the United States, had a lot of older people that were really, affected and died? Were there some areas of the world, possibly in the tropics or other areas, that didn't, this would be dissemination, weren't affected as bad?
So if you look at the data and you run the analyses on the data, it's going to tell you, yes, that there was less, they were less affected. If you work with citizen scientists, the first thing you're going to do is challenge that data. Because for instance, in Austria, where we have a fantastic resolution of who was affected, one of the things that they did during COVID was they set up sampling booths all over the city, all over the country.
Right.
Anybody could go there free of charge, stick a swab up your nose, give it to the person. In 24 hours, the results were on your phone and downloadable. Now, if you want to install something like this in, I don't know, Congo, the Democratic Republic, or South Africa, or Sub-Saharan Africa, anywhere, or Southeast Asia, I mean, I challenge anybody to try to do that. And so in this point, I would just say, that's what the data is showing, but is the data trustworthy? And I don't think so. I just think that all the cases who were probably more affected because of, more widespread poverty, because of lack of infrastructure, because of lack of healthcare services, it just wasn't caught because there was no infrastructure to deliver care to the people, let alone to gather scientific samples that will then be analyzed somewhere. I just think it's a blind spot. And the blind spot now, because it's white, appears like there is nothing happening there. But it's just, what's not happening is data collection, more than anything.
I have one more question.
Yeah.
On tick-borne viruses.
Mhm.
Are you very familiar with
No.
viruses? I had a student contact me about 2 months ago when she was at the Kansas Medical Center, and she had a student a patient come in from Western Kansas, and she asked me, "What do you know about urban tick virus?" Because they had that person come in that died with that, and I said, "I don't know anything about it." And I'm just wondering, have you heard about that?
Yes, I heard about the person who died.
Yeah,
I heard about it.
fatality was in Kansas.
Yeah, but I would lie if I said I was very familiar with the virus. What I would say in this case, though, and I really don't want to, I hate being the Cassandra in these situations. But if you understand the distribution differences between pathogens and disease, then you understand that once the disease starts showing up, we like to trick ourselves into believing that epidemiology tracks diseases real time. It does not, by far. Way behind, because again, with the reverse engineering, we track the disease, not the pathogen, right? This is exactly what we're trying to change with this prevent, with the 3P framework. And once you have a manifestation, you got real lucky because the very first person who got this infection had access to healthcare, decided to go to the doctor, right? And it was a fluke incident, or there have been a multitude of undiagnosed deaths related with similar symptoms. And this one got caught because the doctor just bothered to go a step further and actually diagnose them.
And you have zero idea of how many asymptomatic cases are
Yeah,
out there because
yeah.
nobody's looking for those.
Yeah, I mean, if you look at patient samples who had died of the flu in 2019, and they kept blood samples from them from autumn of 2019 in the United States, and they tested positive for SARS-CoV-2.
Right.
Yeah.
So for instance, one of the things that has been, that came out in our book is that the distribution of pathogens is always much greater than the emergence of the pathogen. And it's called body space. And so it's an interesting idea.
Well, when I was here in the autumn of 2019.
Yeah.
For the Centennial Council, I actually gave a talk here in
Downstairs.
the museum, an evening talk about this. And I made this outrageous statement that the danger was great, the time was short, and we were unprepared. And someone from the medical school in Omaha was outraged at this, and she got up and she was really, really angry, because after all, they had just spent $10 million trying to teach general practitioners in Nebraska to ask patients if they've been in the tropics recently.
Yeah.
That was their contribution. I said, look, you've got, armadillos are already in Nebraska, which means Chagas disease is here. You're going to start seeing people who've never left the state of Nebraska who are now manifesting with a tropical disease that no family practitioner in the state of Nebraska has any idea about.
Yep.
How are you going to figure this stuff? Well, they stomped out. I mean,
Yeah, I wish
The
I
problem was
I
that-
wanted to get her name. So I
yeah,
could
she never
consider
written me to apologize. In retrospect.
In
Follow up.
2020.
But part of what made me so assertive at that point was that we already knew, and this actually gets to something that's relevant for you. We already knew through young colleagues in Hungary who were in contact with young colleagues in China that there was an outbreak in Wuhan that sounded like SARS. And those people they spent the entire autumn of 2019 trying to convince the World Health Organization, the CDC and so on, that we had to stop air travel in and out of Wuhan right then, or this would go global by Christmas. And now, X number of years later, no single public health organization has changed any policies as a result of the COVID outbreak. And there are now, one of the other articles that our group has published is something that the data that Walter picked up. There are now, as a result of our mismanagement, there are more COVID variants in non-humans around the planet than there are in humans. And there 80% of the white-tailed deer in North America carry some version of the Delta variant. And so we've mismanaged something that was 100% preventable into a global example of pathogen pollution that's never going to go away. And it's going to be another one of these background diseases. As far as tick-borne stuff is concerned, there is a group, and I can give you, if you're interested in following this up, I'll give you Gabor's name and email address. There is an EU-funded, what's called a cost project, to establish a ticks and tick-borne disease surveillance network throughout the EU. And one of the most amazing examples of academic scientific cooperation is the people who work with ticks and tick-borne diseases. I mean, they share everything. It's astonishing. They're in contact with everybody. There's none of this is my stuff. This is going to be a big mess.
And just to follow up, like similar to the tick-borne diseases, right? So the tropical versus non-tropical disease, this even when it's a vector-borne disease is oftentimes a great misunderstanding because it's not the virus that's tropical. I mean, viruses couldn't care less what climate they're on. It's the vector that follows a certain distribution. And so now what's happened in 2014 was when chikungunya moved from the Aedes aegypti, which is its tropical vector mosquito, to the Aedes albopictus. And the albopictus is distributed across all of Europe, almost all the way to like the southern parts of Scandinavian countries.
Right.
So then they now have endemic chikungunya populations in southern France, northern Italy. And then in 2019 was the year that they had described the first endemic Zika case linked to an albopictus population also in Europe. right? No travel history, nothing like that. Now, what is worth noting from the Aegypti, which is the tropical vector, is that it carries chikungunya, which is now established in Europe in Diablo pictus. It carries Zika, which is also now established in Europe in Diablo pictus. It carries dengue, and it carries yellow fever. So of the four brothers, two of them have already moved into Diablo pictus. And it's another project that we're kind of discussing because it's a whole different, transdisciplinary speaking, it's a whole different community, whole different stakeholders, different interests, different everything. But scientifically speaking, it's not a question of if we find the other two in the same alternate vector in Europe, it's a when. And the when is going to depend on whether or not we can establish connections with the stakeholders who are constantly exposed to these things, which would be rural communities, farmers, hunters, who spend a lot of time outside in mosquito-ridden areas.
And urban people who are out hiking around.
And urban,
And
yeah,
no longer know to check themselves for ticks
yeah,
when they come home.
yeah.
You know, the first thing you learn as a Boy Scout or a Girl Scout, you know, check yourself for ticks. Nobody knows that anymore.
And that's an issue because, you know, with the ticks, you can say that there are these preventive measures, right? Tuck your pants into your socks. With a mosquito,
Yeah.
I mean, you either put on a hazmat suit or you're gonna get bit. Like there's nothing you can do against them. So if there's an albopictus population, for instance, one of the registered albopictus populations in Vienna is right next to the Schwehat airport, the international airport. And I mean, it doesn't take a lot of imagination to figure out how a mosquito can bite. Yeah. And at this point, it doesn't even have to be a tropical traveler. It can be someone from southern France.
Well, about 5 years ago, one of our colleagues in Vienna, one of the tick people in Vienna, this was the year, it was about 5 years ago that they predicted, very successfully predicted that this summer was going to be in the Northern Hemisphere was going to be a big tick outbreak somewhere. And they knew it was coming. And in Austria, the guy is a faculty member at the veterinary university. He said, we need to put up extra signs, warning signs, and cautionary signs in all the national parks in Austria. And he was called into his dean's office and he was told, you cannot talk about this. If you mention any of this publicly, you will be fired. Because the Ministry of Tourism has said, we cannot risk the economic fallout of making people afraid of ticks in our parks.
And it's very similar situation. So we now in Austria have endemic leishmaniasis cases among farmers with no travel history, no nothing. And one of my colleagues, she is an advisor for the ECDC. She works on eukaryotic parasites, right? So single cell parasites. And she said that it had moved from its original vector to midges. It's in midges.
Oh, yeah.
And over there, leishmaniasis in Austria is a non-notifiable disease.
Right.
Because it's a tropical
It's a
disease.
tropical disease.
It's unknown. But more importantly, if it were notifiable, there is a ginormous lobby of farmers pushing against it. And this is an interesting situation because they're the ones who are infected. Like the very few cases we had came out of farmers working directly with cows. And they're the ones pushing against, or who would be pushing against this legislation. Because immediately, if it becomes notifiable and a cattle is diagnosed or some livestock is diagnosed, they'd have to cull it. And they, because of the way they make their living out of this, right, out of farming, they would rather risk infection than risk having to cull their animals. And I think, and again, like, I can't stress this enough, but this is why it's so important to go eye level with the people who are infected for everybody, because I completely agree with them. I completely see where they're coming from. Of course, I can look at it from a different perspective because I don't make my living off of cattle, but if I did, and I had to feed my kids, then I have a 5%, 1% chance of contracting this disease. Versus if they find some weird scratch on my cow, they're gonna come in and get rid of all 50 of my cows. And what am I gonna do? So it's, it's a very reasonable way of thinking. And this is where you really have to come to eye level and understand what the community has to say. Because I feel like a lot of times the science communication done during COVID was very patronizing. Science is here to tell you how to live your life. Science is here to tell you how to do things. And science doesn't understand a lot of these things, right? And in this case, they're absolutely correct to protect their livelihood because, yeah, if we pass this and we call their cows, good, leishmaniasis is going to be stopped. Then we can figure out how to take care of the unemployed farmers who now don't have anything to make a living.
It's not gonna be stopped anyway, because it's in the rodents.
Well,
So
yeah.
It doesn't make any difference.
Yeah.
Once
Yeah.
it starts showing up in humans, it's so well entrenched in
Yeah.
non-humans.
Yeah.
Yeah,
Doesn't make any difference.
There was an additional thing with COVID and in Hungary was February of 2022, there was a big spike in deaths among elderly Hungarians. And it turned out that the reason for that was because the Hungarian government said that if you were in a town of fewer than 10,000 people, you didn't have to do masking and social distancing. So what happened was at that particular point in time, because kids were generally asymptomatic, they weren't being tested. So they were going to school, they were infecting everybody. Then they were going home, they were infecting their parents. The parents, unless they were deathly ill, the parents were not getting tested because if they tested positive, they couldn't go to work. And they weren't getting paid. Okay. So the government then says if you live in a town of less than 10,000 people, you don't have to mask and social distance. So you now have these asymptomatic kids and marginally symptomatic parents who say, oh, this is great. Let's go out to the village and visit grandma and grandpa.
Yeah.
And so then in February of this big spike of older rural people died because their relatives were killing them as a result of the government policy based on an absence of information.
and I think this is also one of like the big lessons learned, if you will, that I don't know if it's been learned, but should be, is that even though prevention and mapping non-symptomatic, human, non-human population costs money, the reason that it's worth investing into this from a public health perspective is because if you try to manage something like COVID or something like the flu or something like any of these pandemics that we have, it is hopeless. You're never going to manage it. It doesn't matter what kind of healthcare services you have, right? You're going to get exactly what I showed on the first slide, right? You're going to think you're super prepared because you have all of this resource available to you. And 13 months later, you're going to be the most affected by this, right? Because once something like this is out there, there is no way you can snap this transmission chain at any point. You can maybe slow down the rate of people dying, but only as much as the economy has reserved in that particular country, because slowing down means you limit all that economic activity that keeps the country going. And you can't do that forever. So we can flatten the curve, which was the saying during COVID, for as long as we have money to spend on it. And as soon as we run out of it, the curve's going to shoot back up. Because this is exactly the parents are not reporting their diseases because they need to make money. And if they're positive, they can't go to work. So I think this is really kind of the long-term consequences. One of the papers I think I showed was that we wrote about this, exactly this. We compared the kind of the economic loss in case of a pandemic that has not been dealt with before, but only kind of managing afterwards, versus the costs you would have to pay for the prevention. And the difference is that if you don't pay for prevention, right? If you don't invest into these preventive strategies, of course you start higher up, right? With more reserves at your disposal. But the fall is so devastating that it's very difficult to recover because it's a plummeting down. And then there's a little rise back up, but then again, so there's like a very big oscillation. Whereas if you invest into prevention, you start lower, but these mitigations are going to be a lot more smooth, right? And I think this is probably one of the important lessons that we should try and learn.
Yeah, that's what we found in the food security paper. One of the things that Valeria showed is that if we continue with business as usual right now, only with respect to emerging diseases in food, so livestock and if we continue with business as usual, within 70 years in the United States, the cost of treating emerging infectious diseases in the food supply will exceed the anticipated GDP of the entire country. And if you assume that the public health costs follow a similar sort of thing, cut that in half and say, okay, so now the tipping point is 35 years. And now you're in the range of 2050, that anticipated tipping point for a lot of climate change threatened. So it's, it's like they were saying, so we're running out of time. We're really not at the highest levels where we look to people to make decisions. There is no indication that there's any movement away from business as usual. Yeah, they're either so poorly trained that they think that what they're doing is the best that possibly could be done, or they're in such denial that they don't care how much they're lying to the public.
But I might add to this, right, that when working with the governmental stakeholders, right, because like so our lab is the one that had done the entire wastewater-based sequencing for SARS in all of Austria. So we're in regular contact with the ministry, the health ministry and the Stadtwien and all of these stakeholders. So one of the things that I think is really important to note is that on a global level, these policies and these recommendations are very rigid, and it's very difficult to change them in any direction because the consequences are so widespread that it's kind of this butterfly effect, right? We change a little bit up there and it just like blasts through because now everyone's going to be acting up on this. Right. So a very good example for this would be the HPV vaccine program, which they had announced in 2013 or 14 or so, with the target numbers of getting 90% of 9 to 20-year-old girls vaccinated within, I think, 10 years or 5 years or so. And everybody got on board. All the countries that were targeted got on board with this to fight cervical cancer, right? So it's the human papillomavirus that causes cervical cancer. Everybody got on board. And within 6 months, it became very obvious that there is no production facility in the world that can produce this amount of vaccines. And they shut it down. And at first it was restrictions, and then and that's when it came in that the only people that the countries were then allowed to vaccinate free of charge would be girls. Then it was only girls of a particular age. Then it was no multi-age cohort. So someone who would be in the right age for the first, but not in the second. Right. So you had to do all these cuts because it was a very real issue from the private sector where the manufacturers said, like, we'll take the contract. We'd love to produce vaccines for you, but this is how long it takes for us to produce one single. Right. We're talking hundreds of millions. And they're like, there's no way this is going to come off the production line in sooner than 10 years or 5 years, right? So there was this kind of issue to it. But then when you trickle these policies down to the regional and the local level, those are in many, many cases very flexible. And they're actually made for be kind of adaptable to circumstances. And those are these local, the practitioners, the GPs, the local public health official who goes and visits the residential homes and stuff like that. I mean, they have a lot of legroom to work with how a policy is being enacted. And I think that's an entry point right there, right? Because I get why the globals are so rigid. I completely get that. But the local and the regional, they're in a lot of cases, they're interested, right? So the nurse working with the schools or the school board, they don't want epidemics at their schools. They don't want sick kids. They don't want the kids to make their parents sick because everybody knows everybody. I mean, they know the parents, they know the families, they know whoever, and they have an interest in keeping them healthy. And they have enough knowledge and policy that they can actually make decisions. They can adjust. Principals can decide to cancel PE lessons because over there, everyone's sweating and panting and using the same locker rooms, right? So within their own jurisdiction, I think there is a lot of legroom. And in this case, this is where I think it's a very strong bottom-up effort that we can emphasize, where if you get these people on board and you utilize this legroom that oftentimes is just there because there's no input from government or science from the bottom level, right? Everything comes from top down. But if we utilize these leg spaces, or these leg rooms, this is then going to be carried up further in time. But this does generate some bi-directional flow, right? Going upwards.
Okay, well, I think we should end it here. Thank you very much.
Yeah, thank you.
I have pizza order. We're gonna meet over in the mansion lab and eat over there.
All right.
So everybody's come over to the lab.
Thank you for your patience. I know I was keeping everyone